aided drug delivery. So please welcome our attendees. I request everyone to please be patient as Dean Sir joins the meeting.
I think, ma'am, sir is busy some right now, so he asked me to take forward. Okay, thank you, ma'am. Thank you so much for taking lead our workshop. On the behalf of School of Medical Aid Sciences, we are sincerely welcome to all the dignitaries, guest speakers, and the participants across the nation and the globally. So in this workshop, we have several sessions. The sessions are divided into four different segments. It will start from the preparation, characterization, optimization of the nanomedicine, and the the seventh session will be based on the QBD approach for the optimization of the nanomedicine, and the third will be based on the regulatory aspect of uh, nanomedicine, and the last one would be your the pharmacological evaluation of the theranostic nanomedicine. So the entire workshop will go in this manner, and based on that, we will have a different our esteemed guest. We will listen then, and we will get their insightful knowledge, and we will understand the things. So may I have permission to introduce ourselves, our guest speaker first.
Yes, sir.
Oh, thank you so much, ma'am. Dear ladies and gentlemen, allow me to introduce our first speaker and today's workshop chairperson, Dr. M.S. Muthu, Associate Professor of Pharmaceutics at the Department of Pharmaceutical Engineering and Technology, Indian Institute of Technology, BHU, Varanasi. Dr. Muthu is the distinguished researcher in the field of nanomedicine, targeted drug delivery, and the theranostic nanomedicine for therapy and imaging. He obtained his PhD from IIT BHU Varanasi and conducted postdoctoral research in cancer nanotechnology at the National University of Singapore. Dr. Muthu is widely recognized for his work in drug delivery, particularly in nanotechnology and theranostic. He has authored over 110 research articles in reputable national and international journals. His research primarily focuses on targeted nanomedicine theranostic drug delivery for various therapeutic areas. Notably, he served as the editor-in-chief for the internationally peer-reviewed journal Research and Review Journal of Pharmacology and Toxicological Studies. He also sits on the editorial boards of Diagnostic and Therapeutics Journal published by the Luminescence Press. Dr. Muthu has earned numerous scientific awards from the both government and non-government organizations, including the Boycott Fellowship, Academic Excellence Award, CREST Award, IMS Research Publication Award, BHU Vice Chancellor Medical Research Award, International Faculty Award, Best Young Scientist Award, and many more. He is also the recipient of Direct DVT Ghanaian Technology Innovation Award in 2017, presented by the President of India for his contribution to nanomedicine research. His research endeavors have secured substantial funding of rupees more than 2.2 crores from various government and non-government organizations, which include SERB, DST, DBT, ICMR, and BIRAC. Dr. Muthu is a member of National Academy of Science, Nasi, India, since 2019 and Fellow of Royal Society of Chemistry. Furthermore, he has developed targeted nanomedicines for breast cancer therapy with reduced systemic toxicity, which has demonstrated efficacy in preclinical studies. Now, without further ado, I would like to invite Dr. MS Muthu to commence the workshop and deliver his lecture. Please, sir.
Very good morning, all of you. So I'm happy to join this workshop with Dr. Rahul Pratap sir. Organizer, thank you for Rahul for introducing me for the nice introduction. And so also all the participants, I wish you will be enjoying this workshop. So let me share my presentation. Is it visible, Rahul?
Yes, sir. It is visible, sir.
So once again, I thank you, Dr. Rahul, organizer, for inviting me, as well as the GD Institute for giving me the opportunity to share my research presentation on nanothermostics. And mainly, I'll be focusing how the platform can be developed and how it will be useful for targeted delivery, how it will be useful for cancer therapy, and parallelly how the imaging can be done as thermostics. So that will be my main focus. So what is nanotechnology? So the nanotechnology task force, it is a part of US FDA, and they define nanotechnology as the application and understanding of matters at the dimensions of roughly 1 to 100 nanometers. So mainly in the nanotechnology, they consider the size from 1 to 100 nanometer. So how we are developing it and what are the applications, so that will be covered. So that is considered as a nanotechnology. It is as per the definition of US FDA. They have the one division, that is the nanotechnology task force. And the nanotechnology has various applications from material science, molecular biology, medicine, physics, chemistry, and engineering. So the application of medicine of nanotechnology is considered as nanomedicine. So the nanotechnology application in medicine will be considered as nanomedicines. So in another way, we can define that is a technology that facilitates the fabrication of devices on nanoscale. So mainly, it can be considered from 1 to 1000 nanometer. So this is the definition, another definition for nanotechnology. So mainly in the nanotechnology, we deal with the nanoparticles, and we can also say that, so mainly what we are using as a nanoparticle, they are the materials. They used to be in the dimensions in the range from 1 to 1000 nanometer. So they are the materials with the dimension of 1 to 1000 nanometer can be considered as nanoparticles. So how this nano word came? So the nano word is derived from Greek word nanos. The meaning is dwarf or small. So mainly we are using the small material for the medical application, that is the nanomedicine, and it comes under the nanotechnology. So the Norio Taniguchi in 1974, so he coined the term of nanotechnology. So initially nanoparticles were there. The coining of this term nanotechnology came in 1974 by Norio Taniguchi. And so we can also say, nanometer is a 1 billionth of meter. So mainly these particles will be in the range of 10 raised to minus 9 meter. So if you compare these particle, so from macro to nanoscale, so macro scale, you can take example of the person of 180 centimeter and orange in 8 centimeter and hand in 5 mm. So mainly we consider as a macro materials or the macro substances. And in the micro, we can see the example of human hair, smallest eye, the people, and E. coli bacteria. So these all used to be in the in micron range. So 8 micron, 10 micron, and 2 micron. So they will come as a micron. In the nano, we can take a good example, that is a cotton candy. So they are also fibers. So the nanofibers, they used to be in size from 3.5 to 100 nanometer in diameter. And DNA used to be in 2 nanometer, and hydrogen atom, it's very small. So 0.5 nanometer. So we can compare from macro to nanoscale. Now, I hope you have understanding of this nanoscale. And another simple example, that is, if you consider the population of India is 100 crore, so each one of us, Indian, will be considered as a nano out of 100 crores of people in India. So another example, so 1 rupee in 1000 crore, we can consider as a 10 raised to minus 9 of rupees. So that will be also considered as a nano. So now I will classify one way of classifying this nanomaterial or nanomedicine in nanoscale. So we can consider it as a different dimensional material from 0D to 3D material. So what is 0D material? 0 dimension material. So the materials which are having all the dimensions. You can see the dimensions of X, Y, Z. So in all the dimensions are less than 100 nanometer. If you take a particle, if it is less than 100 nanometer in all the dimension X, Y, Z, so they are considered as a 0D material. So they are within the limit of 100. So you can see here one sphere is shown. So all the dimensions are less than 100. So we can take example of quantum dots, nano dots, micelles, and dendrimers. So they all are below 100 nanometer. So they are considered as a 0D materials or 0D nanoscale materials. Next is 1D material. What is 1D material? So materials having 2 dimensions. You can see the 2 dimensions. So yeah, the height and width you can see. Okay, so length is here, right? The length is out of 100. So 2 dimensions are below 100. So they are considered as a 1D material. For example, nanotubes and nanofibers, generally they are very lengthy. So you can see they used to be larger in diameter in one side. So they will be considered as a 1D material. 2 dimensions are below 100. So it's in the 3 axes. So these fiber-based or tubular materials will be used to be lengthy, and one axis will be larger, another 2 axes will be less than 100 nanometer. So they are considered as 1D materials. Now what is 2D material? So 1 dimension will be below 100, and you can see here width and height, they are larger than 100, and so one side is below 100. So you can take an example of nanofilms and graphenes. So 1 dimension is smaller than 100, other 2 are larger than 100. So they are 2D materials. What is 3D material? Similarly, all the 3 dimensions will be larger than 100. So what we use in pharmaceutical mostly, so we will not take a limit of below 100. We always go beyond 100 nanometer also. So like the polymeric nanoparticle liposomes. So the liposomes available in market, that is a doxil, the size is always in 200 to 300. So we use the nanomedicine of also materials which are having dimension more than 100 in all axes. So they are 3-dimensional material. So mainly in pharmaceutical, highly we use a 3-dimensional. So what we see now in the discussion, that is the classification of nanomaterial based on the dimensions. It can be a 0D, 1D, 2D, and 3D materials. So why nano is good? So nothing but why small is good. So the reason is when you reduce the size, so surface area will be getting increased. You can see here surface area is getting increased with the smaller particles. So these particles are having different cells. The smaller particles having 1 cell, and next 2 cells, 3 cells, 4 cells, and 5 cells. It has a different number of atoms. So here the first one having 13 atoms, second one having 55 atoms, third one 147, fourth one 309, and the last one 5th. There are 5 cells and the number of atoms are 561. Now if you compare them, and so the atomic exposure, so the atoms are exposed to the surface 92% with the smaller clusters, and when they are going larger, so the number of exposure of the atoms towards the periphery is less. You can see, so it is getting reduced. So all the particles are more. So most of the particles are getting hidden inside. So they are not exposed to available on the surface for interest. So the percentage of atoms available in particles are smaller. The number of atoms are less in smaller particles, only 13, but their availability for interactions are huge, right? Up to 92% of atoms are available for interaction. So the nano gives better performance in comparison to larger particles. So they may be faster in interaction, lighter in weight, and they are cheaper, and they are in the nanotechnology. So they have many energy applications, and they have different properties at the scale. So the property gets changes. So when the size is getting reduced, and they can be accommodated in the smaller spaces. So in general, in the nanomaterial or nanotechnology, this particular word will come. So when you make a nanoparticle, surface to volume ratio is getting increased. So you can take it here. There is a one cube having 6 surfaces, 6 sides. And if you reduce the same cubes into 8 smaller cubes, so you can see here the overall volume will be same, but the surface area is getting increased. So you can see the height, width, and the number of sides and number of cubes is the surface area formula. If you multiply it, and you will get the 24 mm, that is the surface area of the larger particle. But at the same volume, you reduce the same cubes into 8 parts, and so now you see the surface area is getting doubled, but the volume remains same. So the surface area to volume area is also getting doubled. That is the advantage we get with the small particles or nanoparticles. And it is clearly saying that when the surface to volume ratio increases, so the greater amount of substances will be coming contact. So now we have huge number of receptors for interaction, and we have better, we may have larger number of surface area for absorption. But how these particles will be benefited? So the smaller particles will have more surface area, as well as they have surface to volume ratio is high. So all the hidden portions, so the surfaces will be getting exposed, so they will have the better interaction, and greater proportion of material is available for interaction, so that overall pharmacokinetics and pharmacological activity will be enhanced. So now I come to the definition of nanomedicine. What is nanomedicine? So the nanomedicine is defined as the medical application of nanotechnology. So as I said in the diagram, so there are nanotechnology has various applications. Medical application of nanotechnology is considered as nanomedicine, so that we can use for diagnosis, treatment, or we can use for therapeutic purpose. And for the direct delivery purpose, always we consider from 1 to 1000. Also in the major nanotechnology field, they consider size limit from 1 to 100, but in direct delivery, we consider up to 1000 nanometer because we are taking diagnostic agent, therapeutic agent inside, and so they may be arranged after adsorption, or maybe chemically conjugated, or they are encapsulated. So we can use them for diagnosis, or for treatment, or for as a therapeutic we can use. So now, if you see the classification, we can classify these nanoparticles or nanomedicine into 2 systems. One is a capsular system, another is a matrix system. So in capsular system, like capsules. So the drug is capsulated inside. So there may be membrane outside. So you can take an example of liposomes. So the bilayer membrane will be outside. The inner core will be aqueous core. So the drugs will be capsulated. So that is a capsular system. Another is the matrix system. We can take an example of polymeric nanoparticles, solid lipid nanoparticles, where the drugs are uniformly dispersed throughout the polymeric matrix. So the types of nanoparticle, capsular and matrix system, we can classify into 2. Rahul?
Yes, sir. Yes, sir. I'm here.
Yeah, screen is not changing.
Okay, I think there is some technical glitch. So I think you have to reshare it, sir.
Yes, I will stop sharing. Sorry for the inconvenience. I will share again. Is it visible now?
Yes, sir. It is visible.
Yes. So next we will see what is the requirement for the surface modification of these nanomaterials. So most of the nanomaterials are nowadays given through IV route. This will be the better route for targeting the site. For example, you can take example of doxil for cancer chemotherapy. They are given through IV route. So these kind of particles, after a certain period, they will be getting eliminated by the immune system or reticular endothelial system. So we need to protect them by amphiphilic coating so that they will be having prolonged circulation after the IV administration. So this process is called prevention of opsonization. So the capture of the particle by reticuloendothelial system is considered as opsonization. So you have to prevent it by amphiphilic coating. So this particular technology is introduced in 1994 by Gruff et al. And so once we coat with amphiphilic copolymers, these polymers will have prolonged circulation after IV administration. So we need to coat this particle with the amphiphilic copolymer. So you can see here, these are the uncoated nanoparticles, and this is showing the pegylated nanoparticles, the amphiphilic polymer coating on the surface of the nanoparticles. So if the particles are not coated, so they will be captured by opsonin proteins and they will bind with the macrophages, and finally they will be getting eliminated through reticuloendothelial system. So at the same time, if the particles are coated with polyethylene glycol or any amphiphilic system, so due to the repulsive force, these opsonin proteins will not be binding at the surface of the nanoparticle. There will not be any absorption. So the absorption process will be prevented. So this particle will not be captured by reticuloendothelial system. So these particles will have longer circulation. So whatever the formulation or nanomedicine available in the market for intravenous administration, they used to have this surface modification with any one of the amphiphilic copolymer. So here some of the structure of uncoated nanoparticles like iron oxide nanoparticle, solid lipid nanoparticle, upconversion nanoparticle, gold nanoparticle, liposomes, so and gold nanoparticles. So if you inject them without coating, they will be getting eliminated. So we need to coat them with a different kind of amphiphilic polymers. So polyethylene glycol, polyethylene oxide phosphate, and polyvinyl pyrrolidine dextran. There are different amphiphilic polymers commonly used to coat this nanoparticle to prevent the opsonization process. So the targeting of molecules to a specific site in the body had been pursued actively ever since the development of this targeting method by Paul Ehrlich. It was first investigated, and he used the term of magic bullets. And before the nanomedicine technology developed, so the magic bullets term was used by the Paul Ehrlich. So we can target the site and treat the disease. So that is called magic bullet. The concept was developed by Paul Ehrlich. So the interest in this concept has been increased significantly in the recent decades with the innovations of nanomedicine. So they combined these both, the targeting as well as nanomedicine. So we can say it as a targeted nanomedicine. So, but the technology was there. Now the innovation of nanomedicine was combined, and now it is become targeted nanomedicine. So this targeting can be achieved by passive and active mode. So what is passive mode? So when we inject this particle will be passively reaching to the organs due to the environment in the particular site. So in the case of tumor, what happens? The tumor has the specific environment like hyperpermeability, rapid vascularization. So sometimes defective cells are there and impaired lymphatic drainage is there. So this nanoparticle get accumulate if they are in the size of 10 to 100 nanometer, even sometimes 200 nanometer size particle also get accumulate in the tumor site. And so this particular phenomenon is called enhanced permeation and retention. So generally the nanomedicine for cancer therapy, this phenomena is taken advantage, and people develop the nanocarrier by passive targeting. These particles will be reaching specifically to tumor with the EPR effect. You can see here the blood vessels. Now in the systemic, the particles are injected, and this is the tumor having the vascularization, and you can see the intercellular gap between two cells. Endothelial cells are more, so these particles, smaller particles, get accumulated in the tumor site. So this particular phenomenon is called enhanced permeation and retention effect. So the huge number of particles get accumulated here. You can see here. So we can take advantage. So these particles passively reach to the sites. So that is one of the advantage we take it with cancer chemotherapy. So in active targeting, we use the specific targeting ligand. So for example, we can take advantage of putting some ligand and target to the receptors, or we can take some antibody and it goes to the antigen surface. So this kind of specific interaction So antibodies such as Herceptin, Maptra, Erbitax, so they can recognize very specifically and target some of the cancer receptor present over the tumor. Or we can use smaller molecules, or we can use various peptides like integrin peptide, alpha V, beta 3 integrin peptide we can use. So, or sometimes we can use also aptamers. So they will be arranged on the surface of the nanoparticle for the interaction. So what advantage we get through these nano carriers, whether it is nano passive or active targeting? You can see the nano without the nano system, simple drug molecules, they diffuse lesser across the membrane, and if they are maybe poorly soluble. If they are poorly soluble drug and encapsulated inside the nanoparticles, and by the endocytosis mechanism, they enter more than the drug alone. And in the case of targeted nanoparticles, so we use the targeting ligands, and they enter more into the cancer sites due to the specific interaction, antigen-antibody reaction, or ligand antigen-antibody interaction, or ligand receptor interaction. So the number of molecules enter into the cancer cell will be more through the when we use as a targeted nanoparticles. So you can see here the lower concentration of drug reaches to the target by simple diffusion method. And when we are taking as a nanoparticle, medium concentration will reach to the target. When we are taking as a receptor-mediated things, you can use the folic acid on the surface of the nanoparticle. They target the folate receptor, and they reach more into the cancer cell. So the higher concentration of the drug reaches to the target. So various characterizations are there. So I'm not going to cover. I'll be quickly skipping these slides. So we need to characterize the particle size, Pauli dispersity of the particles, zeta potential, how much drug is encapsulated or entered, how much percentage of drug inside the particles, what is the shape and morphology, and how to characterize them in the in vitro. So these are the how we do particle size. It can be done by microscopical method or light scattering method. And in the light scattering, mainly we use the particle size analysis. So where we will get based on the light intensity fluctuation, laser light intensity fluctuation, we used to get the size of the particle. And through the Pauli dispersity index, we will come to know how these particles will be useful and how they are in distribution. So whether they are uniformly distributed in the same range or in the different range, you can see here it is a highly monodispersed, it is showing. And if the values are 0.1, they are highly monodispersed. If they are 0.1 to 0.4, they used to be moderately polydispersed. If they are 0.4, they are highly polydispersed. And here it is showing the single distribution, and here the curve shows multiple distribution of all the particles within the single suspension. And we need to know the zeta potential for the stability. If they are more charged, positive or negative, they will be highly stable. And so we need to check the entrapment or encapsulation, how much quantity is inside with respect to the polymer content. That is also represented by the formula, and how much drug is loaded. That is can be also calculated by weight per volume. And how to do the in vitro techniques. So there are three methods: membrane diffusion method, sample separation method, and continuous flow method. So the commonly used method, we take the particle inside the dialysis membrane, and the particles will be releasing the drug inside the dialysis membrane. That drug will be diffusing out into the in vitro release medium. So we take the sample out from the outside, and we quantify that how much quantity of the drug is released with respect to time. Now I am coming to the major research discussion part. So the question is there that why cancer could not be cancer so far. So the reasons are there are plenty of reasons. First reason is the cancer is not a one-time event. And you can see here there are different types of cancer. So breast cancer, lung cancer, and different types of cancer. And even in this, there are different varieties. Molecular level, if you see, there are based on the receptor expression, further they are subdivided, and sometimes they are resistant varieties. And the patients are there in the different stages from 0 to 4. So the stage 4 is a very risky condition. The patient used to be in metastasis. You can see the carcinoma 2 metastasis stage. So people used to be there in a different stage, and there are different types of cancer. And sometimes the patients are in chronic condition, and if you start with the treatment is not working out, and they are non-specific in nature. So they also kill the normal cells. There are different resistant varieties of it. So it is not a simple cancer is not a one disease. Simply we can treat with some drug. Even plenty of drugs It is very risky because of the number of stages and number of varieties. So commonly, the people go with the surgery in combination with various therapies in cancer, and generally it may not give the good response due to the resistance and metastatic stage, and sometimes the cancer cells are getting mutated. So now the answer we have to give how cancer can be cured. So the one way is it should be diagnosed at early stage, and we need to start the therapy, first line therapy very early, and continuously it should be monitored, and it should be very targeted. If you are talking about cancer targeted delivery, so they will not be killing the normal cells. So the targeting should be very specific, and subsequent treatments or options should be there, so that will give the better curing option for cancer. So what I'm going to discuss for another 10 to 15 minutes is that, so what are the advanced nano medicine platforms so far we developed in our lab. And now I'll be discussing mainly about some of the micelles and polymeric nanoparticles, mostly institutional nanoparticles for the discussion, which were published from our lab between 2012 to 23. So we identify some of the issues, and we try to solve it. We improved the solubility, so we improved the permeation, and we made some of the targeted drug delivery to breast and lung cancer. We also developed the smart nanomedicine, where the nanoparticles will be delivered very specifically based on the smart behavior, based on the pH and redox environment. And some articles we published, research work we carried out, how they are biodistributive, as well as we also worked on dual targeting based nanomedicine. And so we also delivered them together, two drugs and in combination, and sometimes drug and imaging agent as a thermostic plug. So I'll be discussing those research work published recently. So I'll be starting with the preparation of micelles from our lab, which was published in 2012. So I was the first author of this work. So we developed the TPGS, vitamin E TPGS based micelles. They are non-ionic surfactant. It has both a part, hydrophobic and hydrophilic. And when we take them in the buffer, phosphate buffer saline with the hydrophobic drug, above the CMC values, these micelles are formed in 20 nanometer in size. We prepared with a different quantity of TPGS and prepared the micelles, and we carried out the in vitro release, where we found that the micelles prepared with 150 milligrams showed the better sustained release. And we also checked for their size, morphology, and we have conducted the uptake study. So you can see here, simply micelles prepared with coumarin-6. You can see here in the red channel, this is the cancer cell nucleus, and it's not showing any uptake. And in the case of micelles loaded with coumarin-6, we are finding that in the green channel, the green signal is there, and in the MERS channel, you'll find there are plenty of micelles were taken up by the cells around the nucleus in the cytoplasm, you can see here. And further, we have carried out cytotoxicity study, and we found the improvement in cytotoxicity values up to 2.5-fold with the micelles. And we also checked the biodistribution, where we found that the micelles were taken up more into brain, and it is going more into the blood. So we thought that why can't we use them? They are also reaching more in brain and lungs. Later on, we taken this micelle and start developing them as a target. With these particular micelle is developed as a passive targeted micelles without any targeting event. And passively they reach. So we thought that, so they are passively reaching to brain and lungs, and later on we put along with the targeting ligands, and we studied. The second work here you can see the similar micelles were prepared, and they were arranged with a targeting ligand on the surface, that is a transferrin, and which was done by one of my PhD scholar in 2016. And we checked the morphology by atomic fluor microscopy, and we did the brain distribution kinetics. There we found that in comparison to passive targeted micelles, active targeted micelles were taken up more into the brain at that time of 2 hours. So in further development, so the similar micelles we prepared prepared another one chemical that is modified the TPGS carboxylic with the sulfurdyl group. So these TPGS SH. So once we assembled them, then they had a property of redox sensitivity. So they will be releasing the drug only in the oxidative enzyme environment. So the cancer cell used to have the glutathione in the cancer cells or in the tumor. So they will be specifically getting released only in the tumor site, and we have characterized them and checked for the size and morphology. We conducted the drug release study, And we conducted the in vitro drug release along with the oxidative enzyme, that is a GSS concentration. Then we found that the sulfatyl group containing micelles were delivering the drug only at the GSA, where the concentration of GSS is more. Further, we have done the cellular uptake study and apoptosis study, where we found that the cytotoxic is enhanced and uptake is more, and apoptosis based cell death is more with the targeted nanoparticles. Further, we conducted the in vivo experiment where we checked for the anti-tumor effect. So we developed the lung cancer in the mice by using the enzyme, by using the chemical benzopyrene. So here the histopathological sections are here for the lung sample. So here you can see this, it shows the normal cell number in the cancer in the lungs. You can see the number of cell counts in the lungs. It's a normal one. So this is the positive control where we develop the lung cancer with the benzopyrene. You can see the number of cells are getting enhanced. You can see the dark spots, the number of cells, nucleus are getting enhanced. It confirms the formation of lung cancer. And further, we treated with the control drug and with the different non-targeted and targeted formulations. And where we found that the targeted formulation recovered the tumor here, and number of cells counts were reduced with the targeted nanoparticles treatment. So here it is shown based on the number of nucleus. So this is the normal mice lung cells, and this is the positive control where the number of cancer nuclei are more, and with the treatment they are getting controlled. And based on the Kalpan-Mayer surveys analysis, the mice also improved with the survival. That is analyzed. You can see here that is with the targeted formulation. The violet one, it shows the survival time is getting increased. In one of our studies, we conducted the dual targeting aspects where we prepared the chitosan nanoparticles and we designed with dual targets. We designed with the folate as well as the cetuximab. So two targeting agents were arranged on the surface of the chitosan nanoparticle, and so these are the advantage of these dual targeted nanoparticles. They will be taken up by the two receptor mediated uptake. Let's see the folate receptor mediated endocytosis. So we found the IC50 value. It was around near 5 microgram per ml, and in the IC50 of EGFR mediated cetuximab 1, it was 7.36. And when we combined them, folate and cetuximab, we got the IC50 value of 0.81 microgram. So it confirmed the synergistic effect of the folate and cetuximab in the drug delivery into the cancer cells. And the paper was published in Carbohydrate Polymer with the index of 10. And we characterized them for morphology, size, and the arrangement of the targeting ligands were confirmed with the XPS. And we also conduct the in vitro drug release at the different pH. It confirms the pH-dependent drug release of the chitosan. So the drug were getting more released in acidic pH. So it will be good for treating the cancer. So if the drug is getting released in acidic pH. And we also conducted the cellular uptake study and apoptosis test, and we found that the targeted and non-targeted formulations effect were improved in comparison to control formulation. And we also conduct the pharmacokinetic study where we found that viability also getting enhanced in comparison to free drug. So the next part I will be discussing some of the theranostic aspect. What is theranostic? So here we will have the both diagnostic and therapeutic moieties in the single platform, so that can be useful in the continuous monitoring of the cancer tumor. So we can design in the combination of, we can choose one nano carrier or nano platform, and we can take a fluorophore and drug and one targeting ligand. In the combination, we can get the theranostic. If you take a drug and diagnostic material, it will become theranostic. If you put them along with one targeting ligand, it will become targeted theranostic system. If you are making them in the nano size, there will be nano theranostics. So in one of our study, we made a similar micelle. We prepared with a theranostic micelle. We prepared with the golden nanocrystal. In another set of system, we prepared along with targeting ligand, that is a transferrin receptor targeted theranostic system. Then we characterize them for particle size, quality specificity, encapsulation efficiency, and we also checked for drug loading. When we characterize these particles with transmission electron microscopy, we confirm the size and morphology, and you can see the micelles are arranged. You can see the smaller dark dots, the gold nanoclusters are arranged on the periphery of micelles. It was further confirmed by the targeting ligands were confirmed by XPS with the nitrogen signal, and we also got the presence of gold in the XPS. We got it in near 90 electron volt. We confirmed the presence of gold on the surface of thermostic micelles. And we did the regularly study. Further, we characterized them for the uptake study, cellular uptake. So gold nanocluster uptake, there are a little uptake you can see in the red signal. And when we take along with the drug, here also we find some red signal showing the uptake of the gold. And when we prepare them with the targeted system, so the uptake of this gold nanocluster is more in comparison with the non-targeted system. And their IC50 values were reduced, and formulation is become more potent with the targeted system. So now I will also introduce what is optical imaging. So optical imaging uses the light and special properties of photons to obtain the detailed images of organs, cells, and even some molecules. We can do it, and we can say it as a bioluminescence mode. We can do this optical imaging, or we can do in the fluorescent mode. So for doing the bioluminescent imaging, we need some fluorescent reporter. So we used in our study, there is the luciferase gene-based enzyme expression is overexpressed, and we use the luciferin as a bioluminescent material that I will be showing in the coming slide. And we also use the fluorescent imaging with we have did with the help of the gold nanocluster. Here, the same micelles, thermostic micelles. So after the injection into the SCID mice, immunodeficient mice, we developed the tumor, and we injected, and we checked the fluorescent property of the gold micelles. Here we found that this is the control gold nanocluster, this is the non-targeted, and this is targeted gold nanocluster based. And these are the organs, these are the tumor. 1 is a tumor, and 2 is the brain, and 3 is the liver, 4 is heart, 5 is spleen, 6 is kidney and lungs. But we found that they are very specifically taken up by the micelles were taken up specifically by the tumor. You can see the contrast in the red contrast, green to red shows that better uptake into the tumor. So the same fluorescent intensity is plotted here in histogram. And further, you can see here, this is the bioluminescent based. So we use the luciferin reporter, and we are showing the tumor here. And after the treatment, you can see here the non-targeted and targeted treatment, the micelles were able to reduce the size of the tumor. You can see here, in comparison to drug control, the tumor size got reduced. This one we did by bioluminescence. Another mode is a fluorescent mode. So it is giving the signal with the help of gold nanocluster. So the mice both are same, and it is matching the location. You can see that tumor was created in the xenograft model, and here you can see the tumors clearly located. So after that, we have harvested the tumor, and it is placed here. You can see that it's matching, and these are the 2 mice are same, and the treatment is also visible, as well as the imaging quality is also visible with the help of theranostic micelles. So this particular work was published in Biomedical Journal within the factor of 15. And in recently in IIT-BHU, we have done this particular study. So we created the lung cancer model with the help of benzopyrene, and we have done optical imaging. Here you can see, so we use the DAD, and you can see over the period of time it is getting declined. The fluorescent quality is getting declined with the DAD control. With the help of nanoparticle and targeted system and dual targeted, you can see finally the intensity. You can see here the last one, the dual targeted nanoparticles with the DAD control. It's very specifically, and so they were over the period of time also they were very strong. Signal was very strong here. And after that, we have harvested the lungs, and you can see here. So the formulation were very specific to the lungs with the help of nano system as well as the targeting based. In the dual targeted, this particular chitters and micelles were very specifically taken up by the lungs. The lower part is the heart, and it is very specific to lungs. So this particular work is published in by Micromolecules in ASIS. So another imaging option is the ultrasound and photoacoustic. This particular instrument is having the combined, the showing in the coming slide, the instrument. This is the instrument. The name of the instrument is Fujifilm from the company Visual Sonics. And we use this particular instrument. So it has the dual imaging quality. One is the ultrasound, and another is the photoacoustic. What is ultrasound? It's commonly we use in the clinics. So it provides the information about the anatomy of the particular organ. And so maybe what happened, it has a transducer which sends a beam of ultrasound into the body. So once the sound waves enter into the body, it will get reflected. And between the boundaries, it gets reflected and it will be again captured by the transducer. And so the reflection quality will be getting different between the fluids and between the soft tissue and hard tissue, or between the bones. It will create the structure, so that you might have seen the ultrasound final image in the clinics. And another thing is it is different from ultrasound, that is the photoacoustic. What is photoacoustic? Photo means light and acoustic means sound. Photo in, sound out. That is the meaning. Here we use some of the endogenous contrast, like we are having the hemoglobin content in our blood. So it creates the oxygenated hemoglobin gives signal in red, and deoxygenated give the blue signal. So it will also create the image, but it generates the contrast. You can see here the red contrast is generated due to the blood. It's a good contrast agent. And here the ultrasound, we will not get any contrast. But in the case of photoacoustic, we can generate the contrast. Now, whenever we use any contrast material, so which can be differentiated within the particular photoacoustic image. So now this particular instrument will give you idea both ultrasound as well as the photoacoustic. So here what happened, it is the difference is the input will be the pulse laser from the transducer, and it will be absorbed by the in vivo system, and there will be a thermal expansion of acoustic wave, and finally image will be getting generated. You can see this particular instrument can be used in the various, checking the various pharmacological activity in brain, heart, cancer, and development biology and others. So this particular model which we use, that is the Visual Sonics. So I'll be discussing some of the research based on this usage of this Visual Sonics instrument. And similarly, we had the model of lung cancer model induced by benzopyrene. And these are the ultrasound images. It is a transverse section. It is clearly showing where the lung is located. It's an ultrasound image. And we use the methylene blue based particles. It is a control dye. It is showing the green signal within the lung area. You can see here. And this is towards the heart. And this is the lung will be placed here. So these are the green signal. We get it from here. It is through nanoparticles. And finally, you can see with the targeted nanoparticle, the green signal observed near the vicinity of lungs were high. And it confirms that the particles are targeted and very specifically reaching to the lung area. In another work, we induced the DMPA-induced breast tumor in the orthotopic in the right or left side of the rat. And then we injected with the palbociclib loaded nanoparticles as non-targeted and targeted, and the distribution was observed. And so here the pharmacological activity, the anti-tumor effect, it is very clear. So this is the normal tumor showing the complete growth of tumor, and this is the drug treated, it is getting slightly decline in the tumor size. And in the case of targeted and non-targeted and targeted, you can see, so the target system able to completely recover the tumor, and the tumor is completely getting vanished. And the next you can see here, this is the vascularity of the tumor. The red signal is showing in this particular area the vascularity of the tumor. The first one is the control. It is showing the growth of the tumor. It is clearly confirmed by the red signal, which confirms the vascularity, highly vascularized. And later in the treatment, it is getting controlled by the drug loaded in nanoparticle. So completely get recovered the tumor, and you can see there is no signal at the end of the 8th day. The 8th day, at the last day, there is no vascularity. The tumor got completely recovered. So finally, I am concluding with some of the highlights with our research finding. So whatever we discussed, that is about the cancer nanomedicine, how it can be for the useful for the better efficacy and better imaging. And so initial article I've discussed how the TPGS micelles were developed for brain targeting, later with targeting ligand. And we also evaluated this micelle as a thermostic system based on the fluorescent as well as the bioluminescent imaging. And some of our recent studies, I have shown that shows the synergistic effect of the targeting ligand and the anti-tumor effect in lungs. And I've also shown some of the results related to the lung distribution, which was confirmed by photoacoustic ultrasound and IOS imaging. And one of our studies also estrogen receptor-based targeting of chikungunya nanoparticle, which showed the targeted efficacy and which was evaluated by photoacoustic. That was only. So these are some of the impact factor publications made from our lab. These are the different awards received group, and so these are some of the credits, and these are some of the projects and these students currently under me, and these are the different projects going on. And finally, I would like to acknowledge various funding agency from IIT BHU, ICMR, SERB, and DBT and BIRAC. So this is our lab members. These are our lab members from, these are 4 PhD scholars, Dr. Kasi, Dr. Vikas, and Dr. Shripriya. And so finally, I would like to thank for the invitation here, and thank you for Rahul for inviting and sharing this particular presentation.
Thank you for the informative lecture, sir. It's now time for a question answer session. If participants have any questions regarding the session, please feel free to ask.
May I ask a question?
Yes, sir.
Yeah, so thank you so much. Nice presentation, Dr. Muthu. I have two questions. First question is about TPGS. Why your group is, means why do you feel so that what chemical attributes TPGS has so that you are able to deliver most of the cargoes into brain? Means why TPGS is able to deliver the cargoes into the brain? What chemical attribute it has?
Okay, so your question is about why TPGS is used for brain targeting, right?
Yeah.
Yeah. So it was one of our article. So the remaining article we have used as a linker to conjugate with the targeting ligand. But what the question you raised, that was related to transferring the targeted delivery of micelle delivery or nanoparticle delivery into the brains. So as a passive targeted, this is the non-ionic surfactant. So we can also prepare similarly micelles with the TNAT and some other surfactants. So the advantages with this particular TPGS is that it has both the hydrophilic part and hydrophobic like other surfactant. But in addition, like a conventional surfactant, this particular surfactant, non-ionic surfactant, is a pre-glycoprotein inhibitor. And so it will be useful in the condition of drug resistance, as well as their HLB value is towards the hydrophobic, not towards the hydrophilic, and so that they are better able to transport across the BBB. So we found that this micellar based system were able to deliver the more hydrophobic drug into. So it was one of our study. Then when we combined with the targeted ligand, then the results were further better. And it's not only reported by us. Nowadays, the TPGS-based research with targeting ligand, you will find hundreds of articles. So this was what you are asking. What we have reported is, I think that 11 years back. Now after that, there are plenty of groups published hundreds of articles with the TPGS. Yeah.
Yeah. So one more question in slide number 43. If you can get this slide number 43.
Yeah, I can. You can specifically ask. I will give you answer.
Yeah. So that you found out that some energy bonding, right?
Yeah, for XPS, X-ray photon spectroscopy.
Yes. Some energy bonding diagram, right?
Yes.
So is it obtained by AFM or something?
No, there is an instrument called X-ray photon spectroscopy.
Okay.
XPS. So it gives a binding energy values. The result will be the binding energy value. It will tell the surface composition. So mainly people use them in the material science, this particular equipment, to tell about the surface feature. It will tell about the organic element as well as the inorganic elements. So carbon, oxygen, nitrogen content on the surface, as well as if any metal is present on the surface of any of the material surface, we can get it, and it will give the signal. So it is a relative conformation. So you can have the two surface. One surface is having carbon less, nitrogen more, another surface is nitrogen signal. So we have to compare the structure, and we can confirm that nitrogen signal getting enhanced, so that something attached on the surface. So it is a surface analysis.
Okay, so by using the surface chemistry, you
Yes.
are analyzing the different bonds energy involved into this.
Yes. So mainly we are using some of the targeting ligand which contain more nitrogen. So we look for the nitrogen atom on the surface.
Okay. Thank you so much, sir. Thank you so much.
Are there any more questions? Okay. Once again, thank you, sir. Now I kindly request Professor Dr. Sahil Arora, Dean, School of Medical and Allied Sciences, to welcome all our guests and attendees of today's workshop. So please welcome our participants.
Yeah, thank you, Dr. MS Muthu. Thank you very much for your nice interactive talk. First of all, let me welcome formally for this particular workshop for all the delegates who are here with me for this conference, for this workshop as such. On behalf of Honorable Pro-Chancellor, Mr. Nipun Goenka, Honorable Vice-Chancellor, Professor B.S. Satyanarayan, distinguished guests, scientific experts, and my colleagues from School of Medical and Allied Sciences, and faculty as delegates for this workshop. Ladies and gentlemen, very good morning and welcome on this scientific event, DST SERB sponsored one-day workshop on pharmaceutical, clinical, and regulatory aspects of dual receptor targeted theranostic nanomedicine for effective and safe treatment and detection of brain cancer. It's a privilege for me to welcome Dr. MS Muthu, who is Associate Professor at Department of Pharmaceutical Engineering, IIT BHU Varanasi. I also welcome Dr. Datta Maroti Pawade from School of Pharmacy and Technology, SVKM, NIMS University, Shirpur, who's going to join for the next session, and Dr. Ashok Gupta, Professor in Department of Pharmacy, Sharda University, Greater Noida, who will be another expert for the day in the afternoon session. Dear delegates for the workshop, learning from the nature and adopting it using scientific process is a human tendency. Well, it's a matter of pride for all of us that science has significantly improvised, especially in the synthesis and characterization of engineered nanoparticles, nanomedicines, or nanodevices for cancer diagnosis as well as treatment. Recent years have witnessed an unprecedented growth in the research, particularly in the areas of pharmaceuticals and medicine. We have seen recently how precisely Dr. Muthu has explained as how nanotechnology can be applied to the medicine for the diagnosis purpose, for the treating purpose, for preventing the disease. Of course, now whatever fantasy we used to have in terms of nanoparticulates, now it has converted into reality. It has increased the interest in the future medical application using nanotechnology. Now, a lot of medicines are coming which are based on the nanoparticulates. And I'm sure that in the recent time, this will be emerging as an essential game player in modern medicine. Indeed, there are so many instances where nanoparticles enable analysis and therapies that cannot be performed otherwise. However, if I say nanoparticles, they are unique, but still they have a lot of environmental and societal challenges, particularly to the toxicity. So friends, I'm sure that while working with these nanoparticles as an theranostic, we must also look into the major concern of environmental toxicity or somehow cellular toxicity. That part always be taken care of while working on these things. This meeting basically aims to share new fundamental knowledge and advanced technologies related to the development of nanotechnology-based solutions, especially to the cancer, right? And let's make it sure that further development of it, understanding of these nanoparticulates based delivery system or diagnostic systems, we are continuously improvising the efficacy of these systems, and clinically we are proving it to be effective agent against cancer, especially in the immunosuppressed patients. I once again welcome you all. Congratulate in advance for the organizing committee, which is headed by Dr. Rahul Pratap Singh, for successfully completing the project on nanoparticulate-based theranostic delivery system for brain cancer and making aware about it to the scientific community, which is associated with us today in this workshop. Once again, I, on behalf of management GD Goenka University, welcome you all, and I'm sure today's session will bring a lot of opportunities for each and every one to work further in this brain cancer research. Thank you very much.
Thank you so much, sir. Now I request Dr. Rahul Pratap Singh to invite and introduce our second speaker, Dr. Datta Maruti Papde.
Thank you, ma'am. And thank you to our Dean sir for enlightening over with the topic and highlighted the issue which we are facing in the current health system, and also appraising the efforts of scientists globally. Thank you so much, sir, for your kind words. And I would like to thanks to Dr. MS Muthu, sir, for bringing us a huge knowledge and sparing with their time. Sir, thanks again.
Thank you, Dr. Baude. Welcome again.
Thank you. With the reference of session 2, it will be delivered by Dr. Maruti Pade. Dear ladies and gentlemen, first of all, I would like to introduce our speaker. I am pleased to introduce our second speaker for today's workshop, Dr. Datta Maruti Pade, Assistant Professor at SVKM and NIMS Deemed to be University, School of Pharmacy and Technology Management, Sirpur, Maharashtra, India. Dr. Pade will deliver a lecture on the topic application of QVD principle and the nanoformulation development. Dr. Pade completed his M.Pharm from Department of Pharmaceutical Engineering and Technology, Indian Institute of Technology, BHU, Varanasi. He received a junior research fellowship from National Mission on Himalaya Study, Ministry of Environment, Forest and Climate Change, Government of India, from August 2018 to July 2020. He holds a PhD in pharmaceutics from Naipur, Guwahati. Dr. Pade has received several awards for his contribution in the field and has numerous publications in his credits. Sir, please proceed with your lecture.
Thank you, sir, for the introduction.
Thank you. Thanks.
Sir, screen is visible, sir?
Yes,
Yes,
sir.
it is visible. It is visible.
Thank you. Thank you, sir, for my kind introduction, Rahul sir, and thank you for the opportunity to present my work related to QBD in the formulation development. So today's topic, I will be talking about the application of QBD principles in nanoformulation development. This will be the flow of my presentation. Briefly, I will talk very briefly. I'll talk about the first about the ICH guidelines. Then I will go to the quality by design principles, then design of experiments, then advantages of QBD, and I will briefly discuss what are the misconceptions about the QBD approach, and then I will present a case study. So whenever we make a formulation for any doses form we make, it is not like we will take the excipient and we will go to a final doses form in one step. Suppose we are making a tablet. If this is the API having color yellow, and we are converting it into a dosage form, then like tablet. So first of all, we'll go for granulation. It is general method. Then we'll go for drying, then mixing, then tablet punching, and then if it is a coated tablet, then we'll go for coating, and then that will be a finished product. We will get a tablet. So whenever making a formulation, we will have n number of steps. So n number of steps have n number of factors that affect the quality of product. Suppose we are doing granulation. In granulation, the speed of rotar, speed of shut, and all these things will matter. So again, some excipients, amounts, what is the concentration of diluent, binder, all these things matters like this. So when you go for any doses from development, that pharmaceutical product involves a series of process. So at each stage, there will be number of factors that affect the quality of product. So where chances of variation in the quality of product are very high. So the number of factors affecting the quality of product, how to understand it, how to collect all the factors that can affect the quality of product. So Ishikawa fishbone diagram will help you in that. So if it is a product, you are making a product, what are the factors that are affecting the quality of product, such as if you see that can be a man, machine, environment, measurement, methods, and materials. So if you are making a product, and what are the factors that can affect the quality of product, that can be easily collected at one place with the help of Ishikawa fishbone diagram. So these are the guidelines such as ICH Q8, ICH Q9, and Q10 does talk about the QBD principles in the development of formulations. And so ICH Q8 R2 guidelines is describe the science risk-based approach for the pharmaceutical product development, and Q9 is mainly describe the systematic processes for the assessment of, for the control and communication and review of quality risk. These ICH Q8 and Q9 guidelines are mainly related with the development and manufacturing stage, whereas the ICH Q10 guideline, it is mainly related to the life cycle management of the product. So these ICH guidelines can be we can read, or we can get will get at ICH websites, and it. So I briefly. So what is QBD approach and what is Q quality by testing approach? This is the old approach, that is quality by testing approach. In quality by testing approach, what will we do? We'll make a product, and after that we will test, and we'll ensure the quality at end of the product. Once product is finished, then only we'll inspect whether product formed or not, or whether if it is product is prepared, that is prepared product is with required quality or not. But in case of quality by design principle, we'll build quality by design. In that, we'll first of all understand product understanding, then process understanding. In quality by testing approach, the process is frozen. Once the product is made, and optimized, there will be no change in the process. There will be no change in the excipient concentration little also. It discourages the changes. Where in this quality by design principle, you can change the concentration and the factors amount inflexible with design space. The quality by testing approach is based on reproducibility, whereas the quality by design approach is focused on robustness. So once we go through the slides, we will understand quality by design principle more detailedly. So quality by design principles are introduced by Professor Juran. So Professor Juran says that the quality does not happen accidentally. It must be planned. Whereas in quality by testing approach, we'll test the quality at the end of the product. Then what is quality? Quality is the suitability of drug product for its intended use. If suppose we made a product, whether this product is useful for intended use or not. So what is quality by design principle? Quality by design principle is, it is a systemic approach to develop any formulation or any analytical method or anything. So that begins with the predefined objectives and emphasis on product and process understanding and process control. It is based on sound science and quality risk management. So we'll understand what is the advantage of predefined objectives and what does it means. Very popularly we have seen that whenever we go for sari purchase, particularly, so that time, most of the time, which sari we have to purchase, it is not defined what color, what quality, how much cost. It is not decided. The the objectives are not predefined. So when you go for they go for the sari purchase, that time they see number of saris, and at the end of the day, they may purchase one sari or they may not purchase one sari. But if you see, suppose any boys go for purchase of any clothes, their shopping will be in 5 minutes. They will their objective are predefined. They'll say, I want a shirt, I want this color, and this price. So they will purchase the color shirt in 5 minutes. So what is the difference in both the case? In quality by design approach, we have seen that the objective is predefined so that time is saved. If you see you are sitting in any room, the electricity will be there, so the electricity will be saved. So and the worker who are employee who is showing you, sorry, his time is saved and your time is saved. So that is the advantage that we will save. So what are the advantage of QBD approach? That is, whenever we develop a product with the help of quality by design principle, we'll get a superior quality product as we are understanding the process more soundly. So whenever, again, as we are saving the time, so reduction in the reach to market. I will not read all the advantages, but I will discuss that is reduction in time to reach to market. As we are understanding process completely, we are understanding the excipients interaction, understanding the process interaction with different excipient. So the quality is overall quality and overall factors as we are understand. So that's why the reduction in time in market reach. So as the product quality is improved, superior quality products are formed. So there will be a subsequently drop in the product recalls and rejects. And these are the steps involved in the quality by design principles. First of all, we will decide what will be the target product profile. Once the target product profile is decided, then we'll go for QTPP, that is quality target product profile. Then once the QTPP is decided, then we go for identification of critical quality attributes. When the critical quality attributes are selected, then we'll go for perform risk assessment. And after risk assessment, we'll go for design space. then control strategy, and life cycle management. In subsequent slide, I will explain each terminology. Then what is the target product profile? In target profile is nothing but is the clinical terms such as what is the drug dependence, what is the pharmacology of drug, in which case it can be taken, what are the contradictions, when should not be taken, that is precautions, overdose, what will happen. These all things is considered in case of target product profile. And this TPP is, if you see in case of formulation development, or then what will be the route of administration, what is the dosage form, dose, patient profile. These all the things will come in TPP. And what is the QTPP? QTPP is a natural extension of TPP. It includes quality characteristics that drug should possess to reproducibly deliver the therapeutic benefit promised in the label. So it is nothing but we have to consider what are the quality characteristics that product should have at the end of the manufacturing. So it will deliver the promised label claim. So in QTPP is related to assay, dosage form, purity, and stability of the label. In case of whenever we make a new product, that is NC, new chemical formulation, we develop a formulation for new chemical entity. That is, we have to develop on the basis of existing knowledge base and preliminary information. But when you make a generic product, in that case, we can make it this from reference listed drug products. So what are the critical? Once the QTPP are developed, then we'll go for critical quality attributes. Critical quality attributes whenever you develop, suppose we are making a tablet. So what will be the critical quality attributes for tablet? That will be disintegration time, dissolution time, hardness. These things will be a critical attributes because these are the parameters that can affect the safety and efficacy of the products. So these will be the critical quality attributes. So critical quality attributes is nothing but the physical, chemical, biological, and microbiological properties or characteristics that can be controlled directly or indirectly to ensure product quality. That is, CQA is generally assumed to be a final product's attribute. And as I earlier said, the CQA are only those which are changed after altering or changing the process parameter or formulation parameters. So how to identify whether a parameter is CQA or not? Suppose if we see in case of tablet, we have considered, we will check the quality of tablet, whether it how much disintegration time, how is dissolution time, how is hardness. Suppose whether dissolution is a quality attributes CQA or not. So for that, what we'll see, if we see a CQA is or not. So if we change the concentration of the product, or if this CQA hardness dissolution is changed, whether it is having effect on safety and efficacy or not. If it is changed, if it doesn't have effect on safety and efficacy, then it is not a CQA. If we change the dissolution time, and if it is have effect on safety and efficacy, then it is a CQA again. But if it is CQA, then again next step we will go. In that we will say whether this CQA is changed by, impacted by the variation in formulation parameters, or that is excipient amount or process variation, then it is a CQA. If it is not, no potential effect on the quality of product by changing the formulation parameter, that is excipient amount and material attributes and process parameter, then it is not a CQA. So once the QTPP are developed and CQA are selected, then we'll go for risk assessment. In that risk assessment, we'll understand what are the factors that affect the quality of the products, such as what are the critical material attributes and critical process parameters. Because whenever, as I in very first slide, I have said that when you make a product formulation, when you make a formulation, there will be n number of processes. So n, as there will be n number of processes, so there will be n number of factors that can affect the quality of product. We can't optimize all the factors, that is process parameters and material attributes. We have to select which are the critical material attributes and critical process parameters that affect the quality of product. So we understand what are the high risk factors that can affect the quality of products. For that, if you suppose we are making a nanoparticle system of PLGA based, so what are the quality of factors that can affect the quality of nanoparticles? So material attributes. In material attributes, there will be two things. One will be aqueous phase and other will be organic phase. In aqueous phase, we will generally take surfactant. So which type of surfactant we are using? Ionic, non-ionic. What will be the concentration? What is the volume we are using? What is the pH of that aqueous phase? That will affect the quality of nanoparticles. Suppose we are increasing the concentration of surfactant, so the nanoparticle size may reduce. We are decreasing the concentration, the nanoparticle size may increase. Whereas, suppose we are using a cationic surfactant, then zeta potential may increase after increasing the concentration of surfactant. In organic phase, we'll take PLGA, that will be solubilized in solvent. Then which type of solvent we are using? If we are using PLGA, which type of PLGA you are using? What is the molecular weight? How much amount you are using? That will affect the quality of nanoparticles. And these are the case, these aqueous phase and organic phase we are taken because these are the material attributes. In this, again, there will be homogenization and drying. These are the process parameter. In this process parameter, what will be the factors that can affect? That is homogenization, is the type, which type you are using, what is the speed, how much duration. Same in drying, what is duration, temperature, pressure. So these are the process parameters that can affect the quality of product. So Ishikawa diagram will give overview what are the factors that can affect the quality of product. So once the products are, what are the factors are affecting the quality of product are taken in Ishikawa phase 1 diagram, then we go for failure mode effect analysis. In failure mode effect analysis, we'll say the product, how can the product can fail, or how can the product can be not found, or the quality of the product may degrade. So how could it fail, and what will be the, if it is fail, what will be the effect? That is, we will understand in failure mode effect analysis. Once the failure mode effect analysis is done, then we'll go for RPN scoring. In RPN scoring, what we'll do, we'll understand what is the severity of the process, how much time it can occur, that is occurrence, and if it is a particular phenomenon is occurred, can it is be detected or not. So what will be the outcome of the risk assessment study? That is, we'll understand what are the critical material attributes and critical process parameters. As I said, whenever we make a nanoparticle or something, then there will be some processes and there will be some excipients. So if suppose we are making a nanoparticle system, so if suppose PLG based we are making, then PLG and that solvent will be, and surfactant will be it may be a critical material attributes. And what homogenization and drying, it may be the critical process parameter, that is CMA and CPP. Then once the critical material attributes and critical process parameters are selected, then we will go for design of experiment. So design of experiment is a systematic approach in which we will do a planned experiment. It's not like we'll take any concentration whether it's common or not. We'll plan what high level, low level, and medium level we'll take, and we'll plan and we'll do experiments. What is the advantage of design of experiment? It provides maximum information from minimum data. It gives some equations, graphs, and that will tell about how the parameters are affecting the quality of the product. It will give complete picture at what concentration it is from low stage to medium and to high stage, high level. How the product concentration or process parameter are affecting to the quality of product. Helps in the designing of design space. Design space I will explain in upcoming slides. Uh, Again, as there will be as we seen in Ishikawa Fishbone diagram, there are n number of factors that are affecting the quality of product. So we will not optimize all the parameters that are affecting the quality of. We will screen out. We'll do screening. That design work experiment will help in the screening out the parameter. Only the factors that are affecting the quality of product significantly. That only will take for optimization. Then will you optimize for the so once the screening of factors is done, then we'll optimize with the help of design of experiment. Those factors which are affecting significantly, those only will 4-5 factors will maximum will optimize. That once the advantage of this applying DO is that it will give robust formulation or robust analytical method. There will be variation will be very less. So what are the stages in DOE? Then DOE, first of all, we will define the problem. If suppose we are to make the product, what will be the problems? Then once the problem is defined, we'll determine the objective. If you want to make the product, so if you want to make a nanoparticle system, so how much size you want, how much zeta potential you want, how much entrapment efficiency you want, what time you want the release of the product. That case on that only first of all we will define that objective, that is we want minimum particle size around 200 nanometer, 100 nanometer. We want zeta potential plus plus 25 millivolt or minus 25 millivolt. Whatever you want we will define earlier only, and then we'll go for brainstorming session. In that we'll understand what are the product that can affect quality. Then we'll draw a Ishikawa fishbone diagram. Then we'll go for risk assessment. And risk assessment, what are the factors that are coming? We'll go for design of experiment. In design of experiment, we'll go for one is for screening and other thing is for optimization. In screening, we'll see the n number of factors. That n number of factors that can affect the quality of product, and then we'll screen out what are the critical factors that can affect the quality of product, and that only will optimize. Once the formulations are made, the data will be experiment will be conducted as per the design of experiment, and that data will be collected after analyzing the formulation, and that formula data will be incorporated in design of experiment software, and that data will be analyzed. Once data will be analyzed, then we will interpret the results, and whether this result how the parameters are affecting the quality of product, whether at what portion it is giving minimum particle size, maximum data potential. All those things we'll see in the form of graphs, in the form of equations, and we will see what are the predicted results, what is the optimized formulation, or what will be the design space we'll get at here. So for optimization, for doing design of experiment, what are the designs are available? Therefore, for screening, there are 4 designs available. That is full factorial design, fractional factorial design, blanket Marmol design, and Taguchi design. These are the designs available for screening of the parameters. So screening will help from n number of factors are available for that are affect the quality of product. From that, what are the factors that are potentially affect or vitally affect or maximum affect the quality of product, that will be screened out with the help of screening design. Then for optimization, there are two designs are available. Two designs are available, that is response surface design and mixture design. Whenever we response surface design, there are in that two designs are again available, that is central composite design and box Pinker design. And mixture design will be used for the mixtures, making a solution form. Then mixture design can be used. Suppose we are making suspension or emulsion, in that case you can use mixture design. And in mixture design, there will be simplex design and simplex centroid design. So at the end of design of experiment, we will get a design space. Design space is nothing but a multi-dimensional combination and interaction of input variables, where all the product, all the parameters meet and give a quality product. It means that in that region, if you any formulation you take and make it parameters you take in that particular range, you will get a quality product. And if it, as per regulatory agencies, if from the design space, if you take any formulation or if you change the formulation concentration or process parameter range, then it will not consider the change. So process analytic, once the design space is chosen, then we'll go for process analytical technology, that is PAT. In the PAT is instrument, in that we will use some process, we will use some instrument that will give result on time. If you are making, suppose if you are making any tablet or any in that tablet, if you are going for granulations, so in granulation, at the moment what is happening while granulation, we will not check the quality at the end of the product, that is once the tablet made and then we'll check the quality. Now if you are doing granulation, then what will happening there, at line or online or inline measurement is done. At line means we'll take the suppose we are doing granulation, we'll take the sample out and we'll analyze at other place, that is online at lines analysis. Then second is online analysis. In that online analysis, we'll analyze sample. Suppose we are doing granulation, from that one tube will be there, the sample is taken in the process and will be analyzed from some tubing. In inline measurement, the sample is analyzed in the same system, that is the process, so it will help in the process understanding. Even before that also we can use PAT. Suppose we are purchasing some excipients, so initially we will see that whatever the excipient we are purchasing, that are with quality or not. So that is the at line, online, inline measurements. Once the process understanding is done, then we'll go for. So this is the what are the advantages of PAT? That is, if you use PAT technology, then it will be more reliable and consistent process will be there. There will be flexibility in scale and equipment. There will be better and faster quality system, and there will be chances of process enhancement as we are doing the process PAT. So what are the instrument or techniques are used in the PAT technology? That is, one is used is NIR or Raman is used for the identification of raw material. For wet granulation, if it is wet granulation, we'll measure the moisture distribution. That NIR will be helpful in that. Then drying, in case of drying, moisture content, the same NIR will be used. If you are doing wet granulations, in that granulation with blending is whatever the blend is formed, it's uniform or not, then that NIR technique will be helpful. If you are doing compressions, so we are taking tablet, in that there will be a compression, we use making tablet. So whether the compression force is proper or not applying, that will be checked by strain gauges, and then content uniformity can be checked by NIR. it will generally whenever we make a tablet, we'll test the quality at the end of the product. When the product is finished product will check. But in case of PAT, whenever you use a PAT technique, in that PAT technique, we'll check whether the formulation, whatever the process we are doing, then only we will check. If suppose some problem is coming granulation or in compression force, then we'll rectify there only. Will not go further. So these are the some examples, such as if you are doing some process such as blending you are doing. So in blending or true process analyzer there, if you apply there only, it will tell whether the that formulation you are making, that process is going good or not. That is this is another example, that is spectral probe NIR analyzer. It is installed in GLAD FBD, that will tell with, and there will be no changes in the instrument. It is just applied on the onto that glass, and it will radiate NIR rays, and it will tell whether the drying is happened or not, if the moisture content is not proper or not, whether blend is improved or not. That will tell by this analyzer. So there are some misconceptions about some QVD. Generally, people say QVD we are doing means you use a particular software, and that means QVD is done. No, QVD means you have to start with QVD development. Not that some people will do optimization, QVD is done. No, QVD is not equal to design of experiment. QVD is not equal to design of space. And sometimes people say it is required from industry, required from regulatory agency. Regulatory agency doesn't say we required QVD. But when they say if you use QVD, we'll give flexibility after, if you can do post-approval manufacturing changes after using QVD approach. But QVD is not equal to design of experiment, or QVD is does not equal to design space, and it is not must from regulatory agency. If regulatory agency says that if you use QVD approach, will give regulatory flexibility. You can do post-approval changes within the design space. So as I finished my first part, now I'll go to the second part, that is a case study. During I was in NIPER Guwahati, during my PhD, we have developed ophthalmic emulsion for topical delivery. We optimize with the help of QVD approach. We use drug cyclosporine, and treatment is for dry eye syndrome. So as we are making nano-sized emulsion, so we choose oil phase, that is castor oil, and chitosan as a stabilizer. In that, castor oil is taken and chitosan is dissolved in acetic acid solution, and both are taken on magnetic stirring. Once this oil phase is prepared, then we'll simultaneously prepare water phase, that is in the Pollux we'll take surfactant glycerin, surfactant prolaxomer and glycerin as a for isotonicity, and we'll make a aqueous phase. Once the aqueous phase is ready, we'll transfer this oil phase to aqueous phase. Then once it is transferred, then we'll do magnetic stirring. Once the magnetic stirring is done, both the phases are mixed, then we'll go for homogenization. And after homogenization for 15,000 RPM for 15 minutes, we'll go for probe sonication. Once the probe, as homogenization will help in the size reduction, to reduce particle size of nano-sized emulsion, we'll use probe sonication. As the product we are used for ophthalmic purpose, so we'll do autoclave, so that product will be sterilized, and then we'll be packed this formulation. So as we are doing three processes like magnetic stirring, homogenization, further size reduction by probe sonication. So the variation in the product will be possible from magnetic stirring, homogenization, and probe sonication. This in magnetic stirring there will be again two parameters, that is magnetic stirring speed and time. How much time? If you are doing homogenization, what will be RPM? What will be the time of homogenization? If you are doing probe sonication, what is the time of probe sonication? How much time you are doing, and what will be the amplitude? So these are the process parameters that can affect the quality of nano-sized emulsion. In that, what are the critical process parameters we have to find out? If you are doing, then in case of excipient, the excipients are castor oil, chitosan, water, polaxomer, and glycerin. So in that castor oil, what are the factors that can affect the quality of product? That is, in case of material attributes are castor oil, chitosan, polaxomer, and glycerin. In that, what are the powder that affect the critically? That will be the critical material attributes. So as we are developing the product, first of developing by QBD approach. So in QBD approach, first of all, we have to decide the QTPP, that is quality target profile. In QBD approach, in case of nano-sized emulsion that is developed for ophthalmic purpose. So what will be the dosage form type? It will be colloidal drug delivery. What will be drug delivery type? That is topical application into eye. What will be the route of administration? That is topical ocular route. What will be dosage strength? What will be the packaging? How will you pack it, and how much we want it stable? So while deciding the CQV, what will be the CQV? That is particle size. We have chosen CQ as mean particle size, fold dispersity index, and theta potential. How we decided whether the formulation impact the, how does it impact the quality of product? Whether it is safe and efficacious. If you change the particle size in large extent, it may affect the safety and efficacy of drug product. If you change the concentration of excipients, it may affect the quality of product. That's why we choose mean particle size, fold dispersity index, and theta potential as a critical quality attributes. So once the critical quality attributes are selected, then we'll go for risk assessment study. In risk assessment study, we will develop, first of all, we'll draw our Ishikawa fishbone diagram. In Ishikawa fishbone diagram, as earlier also told, that it will help to what are the all factors that can affect the quality of product. Like in case of nano-sized emulsion, what are the process parameters? Am I audible?
Yes, sir.
So in case of ophthalmic emulsion, what are the material attributes and process parameters that can affect the quality of product? In case of material attributes, there will be castor oil, what type of castor oil you are using, what type of polaxomer you are using, what is the chitosan solution that you are using. These all things will be taken as considered as the parameters that can affect the quality of products. Then how will do final characterization? Then what are the process parameters that can affect the quality of product? That is earlier I told homogenization time. In homogenization type, how much time you are keeping? If you keep for long time, that may reduce the particle size. But if you keep more longer time, it may generate the heat, and that heat may cause adverse effect on the quality of product. You can same as in case of speed. Then what are the environmental factors, that is relative humidity, temperature. Then in case of probe sonication, how much amplitude you are keeping, how much time you are doing probe sonication. If you are doing stirring, then how much speed is there, and how much time you are doing stirring. These are the process parameters that can affect the quality of product. Whereas also, if you are saying any product you are making, there will be always a person will make. So that person's how is understanding it, how it is, whether he is experienced or not, that also affect the quality of product. From all these parameters, these all will be the process parameters and material attributes. Among these, which are the critical? How to find out? So in that for finding out, we'll do first relative risk metric analysis. In relative risk matrix analysis, will whether the any formulation parameter is there or any process parameter is there, will give priority that is high risk, medium risk, and low risk will give. Suppose we are taking a amount of chitosan. If you see chitosan amount is related to, it will affect the zeta potential highly, whereas it is polydispacity, it will have low potential, whereas in mean particle size it is a medium effect. So all these factors will see whether how much they are affecting the quality of product. Then we'll go for risk analysis, and risk analysis will go for risk prioritization scoring. In risk prioritization scoring, earlier told, if any event is occurring, whether it is detectable or not. If it is detectable easily, then we'll go give scoring 1. If it is moderately detected, then we'll give scoring 5. If it is that particular phenomenon is hard to detect, then we'll give scoring 10. Likewise, if it occurrence, if a particular event is occurring, it is unlikely to occur, then we'll give scoring 1. There are 50% chances, 50 chances that can occur, then we'll give scoring 5. And then if it is occurring likely to occur, then we'll give score 10. If it is, and what will be severity? If parameters changed, there will be no effect, then we'll go scoring 1. And if it moderately affect, then we'll give scoring 5. And if it is very severe, then we'll give effect score 10. Then while calculation of RPM scoring, we'll go do the multiplication of detectability into occurrence into severity. In case of nanosized emulsion, as we are preparing, these are the factors we have chosen after the Ishikawa diagram. As this, we give scoring for 5 in case of detection. If it is volume of reagent changed, chances of detection are moderate. That's why you give scoring 5. In case of occurrence is 5, and severity we give 8. So accordingly, with the multiplication and our RPM scoring is generated. And after the RPM scoring generation, we'll go for drawing graphs. That is failure mode effectiveness. The graph is drawn, then the factors which having RPM scoring more than 100, these are the most critical factors that can affect the quality of product. So after once the risk analysis done, then we'll go for factor screening. In case of our design, we use Taguchi design for the factor screening. In that, we use Taguchi design. We taken these 7 factors, that is castor oil, chitosan, poloxamol, bleaching time, homogenization speed, and production time, and then we done that design of experiment give these all 8 runs, and accordingly all 8 runs we have made the formulation, and the formulation are analyzed, and after the analysis all the data we kept in design of experiment, and design experiment will tell which are the factors that can be critical. So this is the Pareto chart we draw. If the that limit is going that particular parameter is going to, about t value, then it is a significant factor. If it is going more beyond the 1 limit, then it is a most significant factor. So accordingly, we have chosen these are the 3 factors that can affect the critically, that is CMA, that is castor oil, chitosan amount, and poloxamol. And only these 3 factors we will now optimize. If you go for all the factor optimize, it will give n number of formulations. The optimization will become very difficult. So for optimization, we used phase central composite design, that is FCDD, and in FCDD there will be 5 levels, and 3 factors we optimize. So we'll optimize, then we'll make a it gave 29 runs. It means we have to make the 29 formulation. We have made the 29 formulation. Then after making the 29 formulation, as dependent variables are mean particle size, Paul-Dissipation index, and zeta potential. So each formulation we have analyzed for mean particle size and zeta potential and Paul-Dissipation index. And this all the data after making the formulation analyzed, after analysis of formulation, we have introduced, incorporated into design of experiment. After design incorporating into design of experiment, it will give some data. On the basis of data, will tell whether the factors are affecting the quality. How does the affecting the quality of product? Suppose we are making considering the one effect, that is mean particle size. So if we see chitosan concentration, if we see as the concentration of the product, that is chitosan, increases, the particle size in curvature effect, that is particle size first of all decreasing and then again increasing and then again decreasing. That particular thing can be observed in case of QBD approach, but this is not possible in case of quality by testing approach, that is QBT approach. So at what moment, at any level, we can tell how does this when any factor is affecting the quality of product. Even we can tell how does it affecting at 6.5 mg, 9.5 mg. These things can be understood by in case of QBD approach, but not in case of quality by testing approach. So once the quality by design approach results are analyzed, then we get the design space. Design space is not a one formula. It is a cluster of formula. It will give n number of formulation. This yellow region is called as design space. In design space, each point is optimized. Each formulation is optimized. It is not like one formulation. It will give bunch of formulation. It will give n number of formulation that can be used as a optimized formulation. So in case of nano-sized emulsion, we can take X1, that is castor oil and chitosan amount, and these are the amount we will take, and we will check how much the particle size is coming. This is the predicted values for particle size, that is 260 nanometer, zeta potential 25 millivolt, PDI 0.27. So these are the predicted values, and then this is given by software. Then we'll take this formula and we'll make formulation, and we'll check whether this formulation, whether these factors are coming matching with the predictor or not. And once it is matched, then we'll say it is optimized. And regulatory agency says that if you take any formula from the design spread test from this yellow region, it will be not considered as a change after post-approval changes. So we have published this article in Farm Scientific. You can get from this journal site. This is the article we published in Farm Scientific. So these are some appreciation moment. I received Best Student Award from Nagpur University, Rashtra Santi Tukaji Maharaj Nagpur. And I received for the QBD approach presentation in received in Control Risk Society first position. Then I got opportunity to present my work in the American Chemical Society student chapter at Bombay College of Pharmacy. So I'm really thankful to Professor Tamilanan sir, who have given me opportunity to learn QBD approach more deeply, also to understand how can QBD approach can be applied to the various formulation and analytical methods. So I'm really thankful to him. Also thankful to Dr. M.S. Muthu sir, because he has taught me how to develop a nano formulation, how to choose any factors to make a nano formulation, how it made, how it is characterized. All this I learned from Muthu sir. And also I'm thankful to my PhD batchmates, Ruhina Rehman and Abhinav Goswami from Naipur Cohorty, because in discussion with them, helped me to go QBD approach more deeply. Also, I'm thankful to my present organization, NMIMS Interview University, for giving me permission to present my work. Also thankful to faculties, deans of GD Goenka University for giving me the opportunity to present my work related to QBD information. I'm also thankful to Dr. Rahul sir for inviting me to give my uh, presentation here. And also, I'm also thankful to all the organizers who are in this presentations. And also thankful to sir. Thank you so much. If anything questions are there, I'm there.
Thank you, sir, for the informative and detailed orientation presentation. If anyone has any questions, please feel free to ask.
May I ask a question?
Yes, sir.
Yes, so, thank you, Dr. Datta, for presentation. I have a question that is it how efficient QVD methods to give that optimum selection of ingredient of a formulation or a component? For example, if we are designing some hydrogel and then we need to select a polymer and cross linkers, right? So how accurate the QVD in terms of helping the researcher to get the best option from a pool of library?
From the pool of library, we can't generally do, we go for risk analysis. In that case, we can do
Suppose we have 5 or 10 types of polymers, right?
Yes.
Or 3 or 4 type of linkers. Okay.
Yes.
So, and that cross linker will help to form hydrogel with the
Yes.
help of polymer, will cross linking the polymer chains, right?
Yes.
Now, can QVD will help us to give that if we want this much of modulus of the hydrogel, we choose this polymer and we choose this type of cross linker. Or else, in more appropriately, we can say that suppose we want to get a biodegradation in normal physiological condition within 21 days. So which type of polymer we should choose for having such specifications? Or can QVD help us for design specification?
Yes, definitely it will help. It will definitely help. We can do in that case, we can take one polymer and one linker, and in that case, what are your critical quality attributes? So we can make some formulation, and what is your target what is your target product profile? Accordingly, whether this product is forming or not. From that, the design expert will give some data about each polymer and each linker. But in that case, generally we don't we use QVD approach after selection of excipient.
Okay.
Generally,
So
generally we will select the excipient first, and then we will go for QVD approach.
okay. So design expert can help into this, right?
Design expert will help you in case of optimization.
Okay.
If you take on any excipient, that product will be how can improve the quality. In that case, it will be more helpful. But in selection of parameters, you can take, suppose you are making some, you can select that case categoric factors.
Okay.
I have taken just factors that have numerical value. You can take n number of polymers. In case of suppose, in case my case, I have optimized that is castor oil, chitosan amount.
Okay.
But in this case, if you can take in this amount, you can take categoric factors. Suppose polymer 1, polymer 2.
Yes.
So
Yes.
how
Yes.
much factor range is coming? From which polymer it is coming good? You can choose by that.
Okay. Thank you, doctor. Thank you so much.
Thank you once again, sir. I would like to invite Dr. Rahul Pratap Singh to introduce Dr. Vikas Jawad as third speaker of our workshop.
Thank you so much, ma'am. Dear ladies and gentlemen, please welcome us our next speaker, Dr. Vikas Jawad, Associate Professor in the Department of Pharmacy at the School of Medical Life Sciences, GTY University. Dr. Jawad is an expert in polymer-based dosage formulation development using novel techniques. He teaches subject as biopharmaceuticals, quality assurance, regulatory affairs, cosmetic science, and industrial pharmacy at GD Goenka University. Before joining GD Goenka University, Dr. Jawad served as an assistant professor at Maharshi Mandeshwar University, where he taught biopharmaceutics and dosage formulation designed to undergraduate students and novel delivery system to master students. He also served as a quality control analyst at various pharmaceutical companies, which include Venus Remedies at Baddi, Himachal Pradesh, and many more, where he tested anti-cancer, antibiotic, analgesic drugs, gaining expertise in various analytical instruments. Dr. Jawad has received the Young Scientist Award in third North Zone Conference sponsored by DBT under the Indian Pharmacological Society. He has a significant number of internationally recognized and peer-reviewed publications, books, and chapters in his career. Sir, please proceed with your lecture.
Sir, please unmute yourself.
Thank you, sir. Rahul sir, for nice introduction. And thank you again, Dr. Rahul Pratap Singh and GD Goenka University for providing this opportunity to present here in this one-day workshop. So let me share my screen first. Sir, my screen is visible?
Yes, sir, it is visible, sir.
Okay, thank you, sir. So before me, since morning, we are having very excellent lectures from Dr. MS Muthu and Dr. Taude, and they have presented very well about the nanotechnology, nanomedicines. But here I'm talking about the regulatory aspects of the nanomedicine. We have different advantages of nanoparticles, nanomedicine as a delivery system to deliver the drug at a specific site and for the treatment of different chronic diseases. But despite of a lot of research since many years, number of formulations based on the nanomedicine are very less in the market. So what could be the reason behind this? I think main reason is the regulatory approval process. So how regulatory approval is affecting the market authorization of the product, and why more number of products are not coming in the market despite a lot of research, we will discuss in the coming slides. So this is the timeline for the nanoparticle and nanomedicine discovery. It started from 1950 to 1960, and till now we are having a different around more than 100 nanomedicine product in the market. But as when we see the research which has been conducted on the nano systems, still the number 100 is very less compared to the research products. So as the nanotechnology and nanomedicine was very well introduced by Dr. Muthu, I think I should skip this slide with the brief introduction only. Nanotechnology is the development of a technology to control the matter at nanoscale and largely at less than 100 nanometer. When we are talking about nano size, we are talking about API as well as drugs which has been processed at nanoscale. And because of the processing at nanoscale, reducing their size, their physicochemical properties got changed, and they may include the physicochemical property, biological, electrical, magnetic, optical, and mechanical properties. Therefore, the extremely minute size provides many benefits in the area of pharmaceuticals and cosmetics to deliver the drug and to increase the efficacy as well as safety of the drugs. However, when we think about the regulatory perspective, still there are many challenges which the scientists throughout the world are facing. Nanomedicine again can be defined as the medicinal, pharmaceutical, or polymeric preparation that can be used to deliver the drug at a defined site, maybe internally or externally, for therapeutic as well as diagnostic purpose. A brief property of material at nano range. When we are talking about nanoparticles, as I told you earlier, there is change in the material at nanoscale. So that change property may be that change in the material may be desirable or undesirable. Sometimes desirable change may include, for example, nanoparticle distribution of nanoparticle in our body. They have access to almost all organs. Their enhanced permeability and retention in the organs will enable us to deliver the drug effectively and for better action. Electronic and optical property of nanoparticles allow them to be used as the diagnostic and as well as imaging material. So when we are talking about nano medicine or nano delivery system, we can have these different types of formulations like liposomes, polymeric nanoparticle, magnetic nanoparticles, micelles, and other different types of nanomaterial with different morphology. The morphological characterization is given here. Okay, so there are different advantages like higher solubility of drugs. Using nanotechnology, nanomedicine, we can improve the solubility of poorly soluble drugs, specifically class 3 and class 4. Then better absorption and higher bioavailability. These are some major advantages which we can get from the nanomedicine. One more important category, that is stability of the drugs, that can be improved by delivering drug through nanomedicine, because when we are talking about nanomedicine, it means we are encapsulating drug in some cavity, some polymeric matrix, so we can avoid the harsh conditions of our body to increase the stability. Then prolonged circulation. As we discussed earlier, even discussed by Muthu sir, the EPR, the permeability as well as absorption and then distribution of the nanoparticles is increased. And because of the nano size, they can distribute throughout the body organs and they will stay in our body for longer duration. Then easy access to all organs and tissues. Nanoparticles can easily cross blood-brain barrier. They can cross placental membrane, and they can be distributed to these organs as well. So it is the advantage of nano particles or nanomedicine to effectively deliver the drug across the body organs. Then targeting approach, as Muthu sir discussed, even in fact, his lab is preparing different targeted nanoparticles. So site-specific delivery is the advantage of nanoparticles, where we can concentrate the drug on a specific site of action. And then delivery of multiple components. When we are talking about nanomedicine, we are delivering different components, maybe diagnostic, maybe therapeutic, maybe theranostic, along within a same entity. So that's why a nanomedicine or nanoparticle could be a multi-component delivery system. For example, as you can see on my screen, a single nanoparticle can have the targeting moieties, surface. We can modify the surface property of the nanoparticle by providing some charge or some ligands to for site-specific delivery, and even to increase the permeability of the nanoparticles. Next is the composition of nanoparticle. We can prepare different types of nanoparticles by providing some coating or by attaching some ligands based on specific purpose. If we are preparing nanoparticles for diagnosing some disease, then we can have gold nanoparticles, iron oxide nanoparticles, and we can make these nanoparticles to site-specific delivery also. And then the physical property, we can have a range of physical properties using these nanoparticles. We can have nanoparticles of different morphological characteristics like rod shape, star-like, triangle, sphere, cubes, and also we can have different porosity nanoparticles. So these are some properties of nanomedicine nanoparticles which we can modify, which we can modulate as per our requirement, as per our the objective. As discussed by Dr. Paude, we should have our objective in our mind, and we can deliver a drug accordingly. So as per our objective, we can modify, we can modulate the modulate the nanoparticle delivery system. Again, this is the example of multi-component delivery system. We can have two parts, theranostic part and therapeutic part and diagnostic part in a same nanoparticle entity. Diagnostic can be magnetic, immunoassay, ultrasound, X-ray, electric imaging, radio, nuclei, fluorescence, optical imaging, and colorimetry. So different types of diagnostic moieties, the diagnostic biomarkers can be attached, can be integrated in a single nanoparticle along with the therapeutic part. Therapeutic part may include the drugs, hormones, genes, antibodies, and the targeting biomarkers as well. Now, there is a big question, how regulatory authorities perceive the nanomedicine? Because we are having different advantages. We can modulate, we can modify nanoparticles as per our objective. Then why nano-based delivery systems are not of so much popular in the market? So there are certain limitations, there are certain regulatory rules which hinder these nanoparticles in the nano delivery system to come into the market. So till now, more than 100 nanoparticles, nanomedicines are available in the market. We can have separate list of these approved drugs from FDA. But the major change that has arisen in the regulatory market after 2017, when the regulatory agencies around the globe realized that they should have some different guidelines for nanosystem. Earlier, they were processing nanosystems as per the conventional system guidelines, but there was a problem. The nanoparticles cannot be approved using or following the conventional system guidelines. And why this is happening? Because of the regulatory guidelines are unclear and non-uniform throughout the world. For the nanosystem, second point, it is very difficult for the innovator to comply the old guidelines for new delivery system. Next, the regulatory authorities, that's why this is the reason regulatory authorities go case by case basis and they approve the nanosystems. For example, one scientist is working on nanoparticle and another one is working on the liposomes. So when we characterize nanoparticle, maybe lipid nanoparticle and then liposomes or some other delivery system from a novel category, they might have their different physical and chemical characterization. So it is very difficult because there are no certain rules for approval of these novel systems. So it would be very difficult for the regulatory agency to approve nanoparticle as well as liposome with the same guideline. So they go on case by case basis. They once they have the case of nanoparticle, they will evaluate, they will ensure the safety of nanoparticle, and then devise the guideline for nanosystem. If someone is applying for liposome or someone is applying for other maybe carbon nanotubes, then they will go with that case. They will ensure the safety of carbon nanotubes and liposome separately, and then they will devise the guideline. So till 2017, they were going like this only, but now they have these separate guidelines. One example is when we are taking blank nanoparticle, it is not a drug. because blank nanoparticle is not having any therapeutic efficacy. So it can be regarded as a device only. And drug molecules are these are the raw material, raw drugs which are having their pharmacological activity. This is a separate entity from nanoparticle. So when we are combining a nanoparticle and drug molecules in a single entity or in combined form, then again this is a problem. Actually, this was a problem for the regulatory authority to define whether it is a device or it is a drug. So, so it was very difficult for the regulatory authority to approve nano system. So, because regulatory authority are having separate guidelines for medical device and for drugs. So, and we are combining here a drug and device. So there is no guideline which for such system till now. Now the morphological aspect, as we have discussed, we can have the different morphologies of the nanosystem, maybe hexagons, triangles, needle-shaped, square, or star-shaped nanosystems. So these morphological differences in the nanosystem again provide difficulty for the approval from the regulatory authority. Because when we are characterizing the nanosystem with different morphologies, definitely it will affect their characterization, their methodology, and then their final optimization. Next one is we can classify the nanomedicine in two categories. Here we are specifically talking about the nano system only. So we can categorize into two categories. One is the medical device. When any implant, when these medical devices are implanted in this in the biological system, they do not have any pharmacological activity, and we can regard them as the inert material. Second is the drugs. When these are injected in the biological system, they will have their function interaction with the cellular moieties, and they will bind to the receptor, and then they will have their pharmacological activity. So this is the difference between medical device and drug. Medical device are not having pharmacological activity, while drugs are having their pharmacological action. Now the third category is the combination product, which we are talking right now. So it is very difficult for the pharmaceutical company to approve such kind of product when any innovator is applying for INDA, NDA, or ANDA, maybe any type of application, depending on the stage of development where they are applying for the new product. So there are certain challenges faced by the product itself and faced by the regulatory authority for the nanomedicine. These challenges can be divided into three categories. Category 1, these are the technical challenges. Category 2, biological challenges, and then regulation related challenges. Technical challenges are the challenges of the product itself. Biological challenges, when we are giving that nano product to the biological system, then what could happen with the body, what could happen with the product? These are included in the biological changes. So first of all, technical challenges. The first problem is the stability of the nanomaterial for scale-up production. Suppose we have optimized the formulation in lab and we are going for the commercial production. So it is very difficult to optimize, very difficult to stabilize the formulation on larger setup, on industrial scale. So if we are not getting that quality, if we are not able to optimize the material, and in fact we are not able to stabilize the material on a larger scale, then it is very difficult to market that product. Second problem, interaction with the reagents at nanoscale. As we discussed, the behavior of material in bulk and in nanoform, it is totally different. So we don't know the interaction of nanomaterial. When I say nanomaterial, it may be an API or it may be an excipient or any other reagent at nano level. So we don't know their interaction with other chemicals, how they are interacting with the reagents of buffer system, excipients, or in fact of our biological system. So we don't know their reaction. So that is the major challenge faced by the nanomedicines, where regulatory fails these products, because we are not able to produce their safety profile. Third point is the morphology of the nanomedicine. As we have discussed here, we are having different morphological aspects of nanomedicine. So because of their change in the morphology, it is very difficult to characterize a single system. Maybe one time we might have a cylindrical nanoparticle, second time we may have oval or circular nanoparticles. So it is very difficult to characterize these particles with the same method, with the same technology. Next one, the manufacturing and reproducibility. Again, manufacturing these nanoparticles on a larger scale and then validation of the process, it is very difficult. One time we are getting the quality product, but there is no surety that second time we will get the same quality. We cannot reproduce the same material following the same process in case of nanomedicine. Another technical challenge is variability in the characterization. Again, for example, the drug content or the entrapment efficiency we find maybe 60% one time. So another time it may be 40% or it may be 80%. So there is a chance of variability of nanomedicine, and this is another technical challenge which any scientist may face during production of medicines. Next one is the lack of analytical technique for nano range. The analytical techniques available for the bulk material may not work for the material at nano range. For example, the optical properties. Optically, bulk material behaves differently than nanomaterial. So it might there are possibilities that the same machine cannot work for nanomaterial which we are using for bulk material. So there are chances of false result, or there are chances that we are not getting the accurate result with the old analytical instrument. Second, deviation from the pharmacokinetic profile of the normal drugs. When we are having drugs in bulk or in conventional dosage form, for example, tablet, capsule, or injectable dosage form, the pharmacokinetic profile of nanomaterial or nanomedicine may deviate from the conventional system. So if the pharmacokinetic profile is deviating, then definitely we will not have the desired bioavailability as well as the desired therapeutic outcome, because different pharmacokinetic profile may lead to toxicity or may lead to subtherapeutic level of the nanosystem. Next one is the difference in the behavior of the nanomedicine in vitro and actual body conditions. This is the biggest problem. We can optimize nanoparticles, nanomedicine in lab. We can have all the parameters within the range. We can encapsulate the significant amount of drug in nanoparticle, but the problem arises when we are injecting these nanoparticles in the biological system, because nanomaterial behaves differently when we are taking in this in vitro conditions and in actual body conditions, because of the interaction of nanomaterial with different body components, biological components. Next challenge, technical challenge, is the difficulty in vitro and in vivo conditions. It is not very convenient, and maybe there are some regulatory norms also. We cannot conduct human trials or animal trials again and again. So for any formulation or any system, we should have some in vitro test that can predict the in vivo performance of the product. So in case of nanomedicine, it is very difficult. Once we establish the in vitro in vivo correlation for conventional system, they work for the coming batches also. But in case of nanomedicine, it is very difficult to establish the in vitro and in vivo conditions correlation. Sorry. Next one is the difficulty in process validation. When we are validating a process of manufacturing, testing, and then transporting nanomedicine system, it is very difficult to validate. Because of the nano size range, we are not getting the desired results every time, and validation process is hindered. Next is the biological challenges. When we talks about the biological challenge, it means after injecting the nanomedicine, nanoparticles inside the biological system, how they are reacting to the body and how body is reacting to those nanosystem is unpredictable. For example, after injecting nanomedicine, there might be some cellular toxicity, organ toxicity, or there might be some allergic reactions instantly after injecting nanomedicine nanoparticles, because our body may take them as a foreign particles. Local toxicity, if we are injecting these nanoparticles as depot formulations. So depot in case of depot formulation, there might be some local toxicity, and there may be some tissue necrosis. Next biological challenge is the drug metabolism. Generally and largely, drugs are metabolized in our liver and then kidney and then some other organs also. But how nanoparticles will interact with the liver enzyme, this is under question. How there is no mechanism to predict how these nanoparticles will react inside the body in case of metabolism. So sometimes excessive metabolism may lead to toxicity of the drug, and sometimes lower metabolism may lead to the subtherapeutic effect of the drug. So in case of nanomedicine, it is unpredictable. Then very known dose dumping. Nanoparticles may release the drug in a single go. So there are chances of dose dumping because of some environmental changes inside the body, pH change, ionic concentration change. So dose dumping could be there because of the dissolution of the polymeric material at nanoscale. Next one is the undesired crossing of blood-brain barrier of nanomedicine. Earlier, we have taken this as an advantage of nanomedicine, that it can easily cross the blood-brain barrier and placenta. But here, again, this is a challenge. Sometimes we want to deliver the drug in liver, but because of their nano size, they are crossing blood-brain barrier. Drug is going directly to the brain by crossing this natural barrier. And there might be some neurotoxicity, there might be some damage to the neurons or other brain disorders. So this is a big question as well as big challenge. How we can prevent nanoparticles going into the brain, and if any pregnant lady is taking these nanoparticles, there are chances that these nanoparticles cross the placenta, and developing fetus may have some abnormality, may have some deformities because of the nanoparticles. So this is a big challenge. Again, undesired systemic accumulation. As we know, the enhanced permeability and retention of the nanoparticles because of the EPR, nanoparticles are going and depositing and storing in our biological system. Their circulation time is increasing. So because of that, there might be some systemic toxicity, unnecessary accumulation of nanoparticles. Then carcinogenicity and genotoxicity. Because of their nano size, minute size, nanoparticles may directly interact with the genetic system, genes, DNA, body proteins, and may hinder their function, and may induce some genetic abnormalities as well as and some diseases like cancer or genotoxicity. Another big challenge is the dose calculation. When we are talking about the conventional system, we have a specific dose for a drug, but in case of nano system, it is very difficult to calculate. It is very difficult to justify the dose with the nano system because of the variation in the physicochemical properties of drug at nanoscale. Dose adjustment is very difficult. And next, biological challenge is unpredictable interaction with the genes, DNA, and biomolecules that we have discussed already. Next one is the regulation-related challenges. There might be a problem in product categorization. As we have discussed earlier, the regulatory authorities feels it's difficult to categorize a nano system in medical device or drug. Earlier, this was a problem, but now they have separate guidelines. It is very easy. So I think now it is not a problem. But again, if we are developing some another novel system which is not available with the regulatory authority, again it will create a problem in product categorization. Next one is the lack of global regulation. Every country is having their own guidelines on nano system. So sometimes they are categorizing one system in drug, another country may have that system in device category. So there is no harmonization of regulations, and this is again creating a problem when an innovator is submitting application for approval in India and in any other country, maybe US. So because of absence of harmonization of regulations, there might be a problem in product approval. Next one is the lack of new product knowledge. As we discussed earlier, when we are talking about new product, it means nanoparticle, liposome, or any other novel system which has not been shown to regulatory authority earlier, has not been submitted to a regulatory authority earlier. So a regulatory authority don't have knowledge of that product to ensure the quality, safety, and efficacy of the medicine. So again, this is very difficult for the regulatory authority to approve the product. So for that, they will demand some extra data, extra studies to ensure the safety and efficacy. Next one is the regulatory authorities demand safety data of bulk material, which does not represent the same material at nanoscale. Currently, different innovators are applying for approval to the regulatory authorities for bulk material, conventional material, for tablets, for capsule, for injectable dosage form. But same thing cannot happen with the nanomedicine, nanoscale, because material is same, but their properties is differing. So old and conventional guidelines cannot approve the nanoscale product. So again, these challenges are given in the flowchart form. The same challenges which has been categorized into technical, biological, and regulatory challenges, these has been described in this flowchart. So there are major regulatory agencies which are involved in approval of nanomedicines are included in, these are included United States, FDA, TGA Australia, CDSCO India, EMA European Union, and then MHRA UK. What are the requirement of regulatory authority to approve any material, including nanomedicines? So regulatory authority wants to ensure their quality, safety, and efficacy of any product, maybe conventional system, maybe nano system. So to signify the quality, any innovator has to submit the CMC, chemistry, manufacturing, and control information to the regulatory authority. For safety, preclinical data is required, and for efficacy, human pharmacokinetic and pharmacodynamic studies and toxicological studies are required to be submitted along with the application, maybe INDA, NDA, BLA, or any like ANDA application. When we are talking about the quality of the product, so description of the nanomaterial should be submitted. It is the requirement of regulatory authority that every product should be described, and their it should be characterized, their required characters like size, charge, surface morphology, and their bonding with the different excipients. It should be provided with the application. Nano material quality attributes. This is the important aspect, as discussed by Dr. Taude also, regarding the quality by design. Critical quality attributes. So this is the point here. Regulatory authority requires CQA. In CQA, they have to provide the critical quality attributes of API as well as the excipients. What are the characteristics, physicochemical properties of the material, and what is the effect of that quality, that characteristic on the final product quality and its performance in the biological system. So here the critical quality attributes are very important parameter that signifies the further quality of the nanomedicine. Then nanomaterial physico-chemical characterization methods. Here, regulatory authority demands what methods and which tests are required to characterize this material. For example, in case of nanomaterial, we may require the particle size, PDI, charge, the stability data, surface functionality of the nano system, and then porosity. All these are the parameters which may affect the performance of the product in the biological system. Next parameter is the dissolution and in vitro release studies. This is the characterization process where we define the drug release pattern and drug release amount with respect to time from any delivery system. So for nano system also, regulatory authority requires in vitro release data. For that, they requires a specific medium, dissolution medium, environmental conditions, maybe the pH of the medium, ionic concentration of the medium. So these are the requirements of regulatory authority which will determine the in vitro release characteristics. And finally, the how drug is released from the material, polymeric material, maybe diffusion, maybe erosion, or maybe the swelling of the material. Next one is the manufacturing process and in-process controls. How we are manufacturing these nanoparticles and how we are controlling different quality parameters during processing of the material, during manufacturing of the material. So this methodology, this process need to be submitted to the regulatory authority. Like, for example, if we are producing nanomaterials, so how we can determine the quality of the nanomaterial produced during in process. So in process quality control parameters are there. We have to take samples during manufacturing, and then we have to check the quality. So as discussed by Dr. Taude, it is a very crucial step where we can check during processing these in-process controls using novel technology, advanced technology, like he has taken the example of fluidized bed dryer. So same can happen with the nanomaterial itself. Next one is the excipients, its types and nature. We are using different types of excipients, specifically polymers in our nano formulation. So regulatory authority requires we have to classify the type of excipient and its role in the formulation. Why we are using a specific excipient? Maybe it is a polymer which is encapsulating the drug. Maybe it is a cross-linker which is polymerizing the polymerizing the polymer to make it in a suitable form, nanoform, or it may be any other excipient. So its type and its role in the formulation should be justified, and it should be with the relevant data. Next is the nature of the excipient. What is nature means? What is the quality? What is the purity of the selected excipient? Why we have taken this excipient? Why it is affecting, whether it is affecting the quality of the formulation final product or not. So all these are the requirement of regulatory about the excipients. Then function of the excipient, safety data. This is the important parameter. As we have discussed, at nanoscale, API may react differently as well as excipient may react differently. When we are taking any excipient, any polymer in bulk, it may not interact with the body. It may be considered as inert material. But in case of nanosystem at nanoscale, that same material cannot be considered as the inert material. So safety profiling of each polymer, each excipient used in the formulation should be provided along with the quality data. And then stability. How we can stabilize our formulation using different polymers and using different container closure system. Because when we are taking material at nanoscale, there are more chances that nanomaterial can react with the packaging material itself. So we have to decide and we have to design the specific container closure system for the nanosystem to stabilize the formulation. storage conditions, and then their shelf life of the product. It should be determined during the data submission. Last is the post-market CMC changes. After approval of the product from the regulatory authority, any innovator, pharmaceutical company can market it. So after approval, they have to collect the data, quality, safety, and efficacy data from the market, performance of the product, and they need to again submit that data to the regulatory authority to ensure the quality, safety, and efficacy of the product for some initial batches only. Next one is the safety of the product. We have determined the quality. Now we have to determine the safety of the product as well. So for safety purpose, any innovator has to conduct preclinical testing on different animal models. Suitable animal model should be selected and should provide the pharmacological, pharmacokinetic, and toxicological data to the regulatory authority. So first one is the dose and exposure. So here in safety data, the innovator should finalize the dose of nanosystem, maximum tolerable dose. For that, LD50 is required, and then exposure time. Exposure time means for how much time we can give that nanomedicine without any side effect. So these studies need to be conducted by the innovator and need to be submitted to the regulatory authority. Next one is the ADME. So complete ADME in animal model should be performed and submitted to the regulatory authority. The pharmacokinetic profiles, its Cmax, Tmax, AUC, and other pharmacokinetic parameters need to be find out during animal study. Now, next one is the most important parameter, the route of administration of nanoparticles or nanosystem. This is very important when we are justifying the safety of the product. Why? When we are taking conventional system, the interaction of conventional system with the biological system is different in comparison to the nanosystem. For example, if we are applying nanoparticles, we are giving nanoparticles through topical route. So they may get absorbed through hair follicles, or the skin pores available, eccrine and apocrine glands. So, and they can deposit under the skin. Now here, when nanoparticles are getting deposited under the skin, they may interact with the sunlight in a different manner as compared to the bulk product. So there might be some allergic reactions with the sunlight when nanoparticles are deposited under the skin. So topical application of nanoparticles might be fatal in some cases. So that's why when we are applying topical, when we are designing and formulating topical nanosystem, it should be characterized accordingly. Subcutaneous administration, when we are giving nanoparticles, nanomedicine in the layers of the skin, it may generate immunogenic reactions in our body. Our body may consider these nanoparticles as the foreign body, and they may react immunogenically. So again, this is a problem. So we have to justify the safety of nanoparticles for subcutaneous administration in that manner. Inhalation, if we are giving nanoparticles through nasal route, so their lung toxicity, their toxicity to the alveoli may be in a different manner as compared to the bulk product. So when we are developing inhalation product, their lung toxicity should be evaluated. Next parameter is the intravenous product. When we are giving nanoparticles through IV route, these may react with different body proteins, blood proteins, body tissue in a different manner. They may get deposited in some area of the body. They may form complex with the blood proteins, and there are high chances that their circulation time will further increase. So again, this is a problem as well as benefit during the safety study of the developed nanomedicine. Oral product. When we are giving nanomedicine through oral route, definitely it is going to increase the solubility as well as absorption through oral route. But again, there might be a problem of toxicity, oral toxicity, because deciding a dose is very difficult when we are comparing nanosystem with the conventional system. Next one is the testing. Testing of the representative nanomaterial for compatibility study. This is the requirement of regulatory agency from the innovator. They ask to conduct the testing of excipients itself on the biological system. When we are developing nanoparticles, drug-loaded nanoparticles, that nanoparticle is having polymer and drug, but regulatory authority requires the biological study of polymer only. So we need to prepare blank nanoparticle, and we have to conduct some biological studies on those blank nanoparticle to represent their safety in the biological system, to represent their safety in case of organ toxicity, in case of crossing of blood-brain barrier, neurotoxicity, nephrotoxicity. So their toxicological profiling of blank nanoparticles is required by the regulatory authority to ensure that the nanomaterial, the excipients at nano range are safe and suitable for making nanomedicine. Next one is the bridging toxicology from drug product on not containing nanomedicine and to drug containing nanomaterial. There is a huge difference in the toxicological profile of material in bulk and drug material in nanosystem, because the interaction of bulk material with the nanosystem as compared with the biological system, and interaction of nanosystem with the biological system is in a different manner. Sometimes nanomaterial may induce toxicity and immunogenic and allergic reactions in body. So that's why toxicological studies of nanosystem should be compared with the bulk system, and it should be submitted to the regulatory authority to ensure their safety in the biological system. Next part is the efficacy of the product. Once we have determined the safety of the product, now next is the assurance of efficacy of the product to the regulatory authority during INDA, ANDA or NDA submission. So they require the product introduction, background of the product. In background, we need to provide processing of the material, manufacturing process and their evaluation criteria, and then clinical development process. How we have conducted the clinical trials of this nanomaterial, and what are the results, what is the final outcome from the clinical studies, and what is the risk to benefit ratio of this newly developed product. It should be submitted to the regulatory authority. Human pharmacokinetics to determine the safety and efficacy of the material. So in efficacy studies, nanoparticle formulation should be given to the human volunteers, maybe healthy volunteers and maybe diseased person, depending on the phase of the clinical trial, and the pharmacokinetic profile should be evaluated, and it should be compared with the conventional system to justify that the nano system is as much efficacious and even in more efficacious than the conventional system. So this is the step where we are justifying nano system as equal or more efficacious to the conventional system. Then toxicological data. Again, a human toxicological study should be provided to the regulatory authority of the nanomaterial. When we are talking about toxicological studies, again, the toxicology of only excipient and toxicology of nanoparticle containing drug, both should be provided to the regulatory authority. Last one is the bioavailability and bioequivalence study. If we are applying for ANDA, abbreviated new drug application, then bioavailability and bioequivalence data should be submitted to the regulatory authority to ensure the efficacy of the product as compared to the innovator product or as compared to the patent expired drugs. So this is how we can apply with the regulatory authority for different nanomedicines to get the market authorization. Again, this is very difficult to get the market authorization of nanomedicines because of the challenges, as we have discussed, maybe technical challenge, biological challenge, or regulatory related challenges. Thank you. That's all from my side.
Sir, thank you so much. for your excellent presentation. Sir, if there are any question regarding the topic, please feel free to contact to ask.
Thank you, Dr. Vikas, for my presentation. I have one question regarding that. You talk about different guidelines for regarding nanomaterials, right?
Yes, sir.
Okay, so I have one question that is there any coordination or harmonization between different regulatory agencies regarding nanomaterials?
Yes, sir.
If yes, and what are those guidelines?
Yes, sir. First of all, every country is having their own guidelines for nanomaterials. Maybe TCA, Australia, CDSCO, India, US FDA, USA, they are having their own guidelines. Now, about the coordination, this is the missing link throughout the world where they need to coordinate again and to harmonize the guidelines. In case of conventional system, there are ICH guidelines which harmonize the quality, safety, and efficacy system throughout the world for conventional medicinal system. But again, in case of nanomedicine, this is the missing link where scientists as well as regulatory authorities around the globe are working on this, and definitely they will have some common guidelines throughout the world. But as of now, every country is having their own guidelines, and generally they are following US FDA guidelines to approve the nanomaterial.
So I think we need something like ICH for nanomaterials also.
Definitely, sir.
Thank you so much.
Sir, I have few questions related to topic, sir.
Yes, sir.
Sir, in my project, we have developed theranostic nanomedicine. We have done preclinical study. Now, what are the steps to move forward and take the approval from the government of India for further process?
Yes, sir. First of all, you have to prepare your data as per the quality, safety, and efficacy profile of the developed product, and then you can apply with the. In case of India, you can apply with the CDSCO for preclinical testing. Again, because in preclinical studies, in educational setup, we might have missing some data. So first of all, you have to apply for IND application with the CDSCO and then move further.
Yes, sir. Sir, thank you so much for your giving a wonderful presentation, your insightful comments. Sir, thank you once again. With reference to next session, dear ladies and gentlemen, we would like to introduce our fourth or the last, not least, our speaker, Professor Ashok Kumar Gupta, sir. He is a professor in the Department of Pharmacy at Sada University, Greater Noida, Uttar Pradesh. Dr. Gupta holds a PhD in pharmacology, which he has done from Institute of Medical Science, Banaras University, Varanasi, and has over 17 years of experience in teaching and research. His research area include quality of life, drug utilization, pharmacovigilance, drug clinical studies, and on the chronic diseases, herbal drug studies on animal models. Dr. Gupta has published more than 30 scopus and SCI index journal paper in international and national recognized journals and holds the 3 granted patents. Sir, please proceed with your lectures.
Thanks, Dr. Rahul, for introducing me, and thanks for the opportunity given to me, to talk in this platform. And I congratulate to you and your team, for organizing this workshop on the very modern era of medicines, that is theranostic use, especially in the brain cancer patients. So thank you for introducing me.
Thank you so much. Thank you.
Now I think the slide is shared with you. It's visible to all.
Yes, slides are visible.
Yes,
Fine.
sir, visible.
Okay, sir. Okay, so my topic of presentation is predictors of outcome in the management of brain cancer. So first, I would like to a little bit discuss about the one kind of cancers that happens and severely affect the human beings in different proportions. So today, the topic is related to solid tumors. Brain cancer is a type of solid tumors. So many other kind of solid tumors are there, that is prostate cancer, breast cancer, The cancers of head and neck regions, so especially the mouth cancer, so many entities are affected with this, and the outcomes are different for each and every cancers. Especially why this brain cancer is so different from other solid tumors, because of heterogeneity, lots of variants are there, and the very delicate area where it appears, that is our brain. That is the network of several neurons which have different functions to regulate different organs of the human body. But why this is dangerous with other solid tumors, if you talk about the prostate cancer or breast cancer, so their area of location is quite different. And the operational procedures are easy for those organs. So here, let us start with the brain cancer, and we'll discuss about the various approaches which is being used or already used and still is the part of clinical trials. And the scientists and various researchers are working on it, and how to correlate these things, okay, for maximize to benefit to the patients, especially for the survival, because the cure is still impossible, especially high grade of brain tumors or brain cancers, especially grade 3 and 4. So we can still increase the survival years for those patients who are already suffering with this brain cancer. So these are the content of my presentation: introduction, brain tumor types and grade staging. We'll talk about the barrier system, that is blood-brain barrier and blood tumor barrier, acting passive targeting, hyperthermia approach, toxin-based approach, convection, enhanced delivery, nucleic acid, and so on. So we'll take the prevalence of cancer. So in India, almost among 9 people, 1 people are most likely to develop cancer in their lifetime. So that is the prevalence in Indian patients. According to Global Cancer Observatory, about 19.3 million people are affected by brain cancer in 2020. And of this 19.3 million, very significant proportion belongs to Asian populations. India among this ranked third after China and America. So it's a huge number. The patient in Indian origin or Asian origin, or even the people from different parts of the world, they are suffering with the brain tumors. The high prevalence of brain cancer in India, it further supposed to increase about 57.5% in 2040. So it's a huge figure. So we must work on how to prevent it, how to cure the patient, how to prolong the survival of those patients who are already suffering with brain tumors. Brain tumors, if you take especially this solid tumor, so it affects across all the ages and it causes significant mortality and morbidity. So morbidity in terms of it causes cognitive decline, mental distortion, and other problems related to mental health. And the people's survival rate is also very less who are suffering with brain tumors. According to global cancer statistics, there are 3 lakh new cases identified with brain or CNS cancers in 2020, and there's a death about 2.5 lakh cancer patients in 2020 worldwide. So it's a huge figure again. So we must look inside how to maximize, how to cure these patients or prolong the survival of those patients. Why this brain cancer or CNS tumors are unique? Because histologically they are complex. There are so many variants of brain tumors available which affects the different part of brain, and because the network of several neurons, so surgery is not an option always if for treatment of these kind of patients. Over 100 types of variants are available as defined by WHO related to brain cancer. If you talk about the grade and types, so brain tumors can be categorized into 2 grades, that is low grade tumors and high grade tumors. Low grade tumors means the grade 1 and 2, and high grade tumors, that is grade 3 and 4. Definitely low grade tumors, the survival is always better, but in high grade tumor, survival is always worst in that situation. So we have to focus in both directions, especially with the high grade tumors, the patient who are already identified with the high grade tumors. Now, if we talk about the types, So there are mainly two types of brain tumors, that is gliomas and non-glial tumors. Among these two kind of tumors, the gliomas are very aggressive kind of tumor, brain cancer, that causes lots of death every year. The patient suffering with each and every age group, okay, or each and every sexual types. Even the children, they are affected with this gliomas, and the death, so many deaths are reported every year. So the gliomas as a leading cause of death related to brain cancer, the males are more affected than women's, and it's more common in white as compared to blacks, as reported by as available in the data. So here, the patient who are suffering with gliomas, the overall survival time for those patients is about 1.25 years. That is very less if you compare it with prostate cancer, breast cancer, or any other solid tumors that happens to human beings. So here the survival rate is very less. So how we can prolong this survival by using this modern era of theranostics or nanomedicine, okay? So combination of all these approaches, conventional medicines as well as the new generation medicines, that should be our aim. If you talk about the different, so there are four grades. I already talked about the grade 1 and 2. It belongs to low grade tumor, and grade 3 and 4. It's a aggressive kind of tumor, especially the grade 4. If you compare with grade 1 and grade 4 treatment options, so definitely there are lots of difference between grade 1 and grade 4 treatment option. Surgery is possible with grade 1, surgery is not possible with grade 4. Only the palliative care can be given to those patients. So our aim is to focus whether what is the age of that patient, which area has affected, because if you look at the brain cancer, it comes to the cerebral cortex, and there are different lobes. Each and every lobe, whether it's if I talk about the frontal lobe, parietal lobe, occipital lobe, or this temporal lobe. So each and every lobe have their very significant functions for the human body. If you lose any portions, so it affects the functioning of different organs, especially the cognitive functions. So we have to look inside. First, we have to define the grades, whether the cancer belongs to which grade, 1, 2, 3, or 4. Especially if it belongs to grade 4, then survival is always very less. Now, this is the staging system mainly used for staging of the solid tumors. So here we are using this, or that is defined by American Joint Committee of Cancer. It's a TNM staging. So what does it mean by TNM? T stands for size of the primary tumor and their location as well. N stands for the lymph node involvement, and M stands for the metastasis. So it's a TNM staging, both pathological as well as clinical staging is made on the basis of this TNM staging system. So T stands for, means T we further categorize as T0 to T4. T0 or T1 means the very small tumor size. T4 means it's a very big size of tumor, and that is represents the lymph node involvement. Okay, it's obvious the first any kind of solid tumor, it first where it spread before metastasis to the blood, it goes inside the lymph nodes or in lymphatic system. So still there is a chance to survive for the survival of those patients if the cancer is detected in that manner. But the extent how much lymph nodes are involved, that must be detected through imaging processes. So lymph node involvement is also as important as the site of tumor that happens inside the brain. So most usual site of lymph node which is involved in brain tumor, that is cervical lymph node, that lymph node which is available in your neck region. So that should be identified and must be screened out these kind of patients, okay, whether metastasis has started or not. Now M represents ultimately the metastasis of this tumor, whether has spread by organs or inside the blood. So this is about the TNM staging, and that must be identified, okay, before treatment, because all the treatment option is based upon this TNM stage. Now, if you talk about sorry for interruptions. Okay. If you talk about the different barrier systems, okay, so there are two kinds of barrier system, that is blood-brain barrier and second is blood-brain tumor barrier. So why this barrier system is important? Because each and every treatment which is given through the blood or systemic route or IV route, okay, so these barrier system stops or interrupt, okay, in that efficacy of those treatment options. Even if you are going for the nanotechnology devices, okay, so that system also always interrupt with those kind of medicines used for the treatment of these kind of cancers. So let us discuss about the triple B and BBTB, okay, means blood-brain barrier and blood tumor barrier. Why this barrier is important? Because whenever the cancer develops, okay, so if you correlate with the grades, whether it belongs to grade 1 or grade 4, so what is the difference between these grades? Okay, because in grade 1, this barrier system is not affected at all or mildly affected. But if we talk about the grade 4, so this barrier system is severely affected and it causes leakage, it affects the penetration, or it causes extra accumulation of those drugs, okay, being accumulated at the site of where tumor has developed. So that part is important. What is the grade of the tumor, okay, and how it affects this barrier system. That is for disease as well as for the treatments options available for this brain cancer. Now, in this situation, the gold and iron oxide based nanoparticles are giving very promising results because they can be modified, okay, they can be moderated, okay, as per use, okay, they can be locked with different kind of agents, anti-cancer agents, okay, and delivered to the required sites. So they are giving lots of options are there, but this gold and iron oxide based nanoparticles are giving very promising results. So theranostic nanoparticles can be used to avoid frequent and invasive dosing and improve patient compliance. So that is the big thing, okay, while using this theranostic nanoparticles, okay, especially designed with gold and iron oxide based options. Now, if we talk about the triple B, so large molecules, as you already know, okay, and it expresses because where this triple B barrier is there, so that is so many expressions are there, so many molecules, exogenous molecules are controlled by this ATP binding cassette, that is a transporter system, and efflux transport proteins. So these two proteins, especially the ATP binding cassettes and efflux transport protein, it controls this endothelial cell membrane transport processes. So we can target, or the scientists, they are targeting these two proteins, okay, for permeability or for increasing the dose regimens or specific dosing towards this brain cancer tissues. Now, other approaches may be transcytosis approach, that is carrier-mediated transcytosis, or receptor-mediated transcytosis, or adaptive-mediated transcytosis approach may be used for this brain cancer targeting. So alternative pathways transferring the triple B have been investigated for their potential application in invasive drug delivery. Now, if we talk about the blood-brain tumor barrier, so why this triple B is different from blood-brain tumor barrier? So that blood-brain tumor barrier is entirely different from this triple B and more complex, because here the tumors grows and it affects the permeability of those, it affects the tight junctions where leakage can happen, and that may responsible for penetration in other tissues, okay, as well as it affects the treatment options. Here, the endothelial cells are very compromised, especially with this blood-brain tumor barrier system, and causes lots of challenges, okay, for the treatment of this brain cancer. Because of this leakage and high metabolic stress, it causes it develops hypoxic areas, okay, that create further problems in the treatment of these kind of tumors. Again, fenestration in this tumor barrier system, okay, it causes significantly higher permeability compared to the triple B, allowing small molecules to pass from the bloodstream into the brain. So fenestration in blood-brain tumor barrier system should be looked out, and we can work on this fenestration system of this to maximize the treatment of this, whatever treatment option is available. Now, the third things which is evident, okay, especially with the brain tumors, in blood-brain tumor barriers, there are so many overexpressed receptors that could be targeted by using ligand binding approach, or means receptor first should be identified, and by ligand binding approach we can target those kind of receptors, okay, for the treatment of this different kind of brain cancer, especially the gliomas. Now, this is the picture depiction of this blood-brain barrier and the tumor barrier. Here, the extra layer depicts the tumor barrier. Okay, so the second picture, the diagram B, is more complex, and it's actually this is the challenge for this treatment. Okay, so because each and every tumors of that happens inside the brain, it has different heterogeneity. They have different expressions. They have different permeability problems. So each and everything must be worked out, okay? And that directly related to the grades of that tumor, whether it belongs to grade 1st or grade 4. Definitely in grade 4s, these kind of challenges are more in compared to grade 1st kind of tumors. Now, there are two kind of approaches being used for this treatment of brain cancer, that is passive targeting and active targeting. Not only for brain cancer, so many other cancers are being treated by using this approach. If you talk about the passive targeting, it's obvious. It's a normal membrane, means their permeability is utilized, okay, being utilized by using especially the lipid carrier system, because the plasma membrane, okay, the lipid-based systems easily crosses. So we can encapsulate those system or liposomes derivatives for the treatment of these kind of cancers. That is passive targeting. There is no specific ligand binding approach works over here. So we can use passive targeting, or the second option is active targeting. Here, it's maybe this second option may be used, okay, or widely used about the treatment of this, because in this situation, the site desired receptors we want to target, that can be ligand by ligand approach, okay, drug ligand approach, okay, we can target those kind of ligands, okay, and we can target especially those kind of tumors, which is, and these active targeting is widely used especially for this grade 4 kind of tumors. Now, one approach which is very popular, that is hyperthermia approach for brain tumor treatments. What is hyperthermia? We are using about the temperature around 43 to 41 degrees centigrade for treatment of brain cancers. This therapy was commonly used especially for the treatment of this prostate cancer. The HIFU therapy, that is high intensity focused ultrasound, that rays are being used for the treatment of prostate cancer patient. The same option is modified and being used for the treatment of this brain cancer patient as well. So hyperthermia approach, okay, could be a possible option for the treatment of brain cancer. Why this hyperthermia? Because it breaks down the tight junctions to promote focused pharmacological delivery and also change the functionality of cellular structures. So hyperthermia and approach may be used with this manner. And second, temperature can be synergistically and selectively kill cancer cells. Okay, so by using the different on the basis of grade of tumors, okay, we can modify or dose of this tools which is used for incident the temperatures, okay, by using this focused ultrasound, radio frequency, microwaves, laser techniques, or magnetic energy, okay, to target those cancer tissues. So it's an option just similar to HIFU treatment, that is high intensity focused ultrasound being used, but still this part is still a part of this clinical trials, because the heat that is being used for treatment of this, okay, that may damage to the normal tissues as well. Now, the next is toxin-based approach for the treatment of brain cancer. This toxin-based approach was identified in 1970s. Here, the toxins produced by the bacteria, fungi, or other microbes, or other sources, okay, being utilized for targeting the brain tissues, brain cancer tissues. So here, immunotoxins, we can say the cytotoxins, okay, is being used for, and their recombinant molecules are being used for, means targeting the cancer cells, okay. So that binds with the antigens, which is overexpressed on the surface of cancer cells. So this kind of therapy is still the part of clinical trials, but we can use in combination with other kind of treatment, okay, for better efficacy or for the survival of those kind of patients. The few toxins being used, okay, already means used in the various studies. They are pseudomonas exotoxin and diphtheria toxins against means the breast brain cancer. Now the approach is this, okay, tox-based treatment. The several receptors are identified like transferrin receptor, interculin receptors, interleukin 13 receptors, epidermal growth factors, and other kind of receptors, okay, which are overexpressed on the surface of this brain tumor cells, which is can be approached by using this toxin-based different toxin-based drugs with combination with other agents, or we can use this toxin-based drugs, okay, as a part to combine with this nanoparticle approach. Now the convection enhanced treatment. So what does it mean, convection enhanced treatment? Okay, that is also adjunctive treatment for the treatment of brain cancer. So as we know that the cancer cells are infiltrative in nature and invade beyond the site of origin. So, and our BBB, that is blood-brain barrier, blocks the intravenous drug from adequate distribution to the regions of cell infiltration. Facing in the treatment of this these kind of cancer. So here, by using this convection options, by bypassing the BBB and infusing drugs directly into the tumor with intracranial catheters. So this is the intracranial catheters that is being used to target or to lock the desired dose of the drug at the desired point or desired site. So that is convection enhanced delivery. So we can use these kind of convection enhanced delivery, okay, for as adjunctive treatments for treatment of this different kind of brain cancers. Now the problem with these kind of convection enhanced delivery is the inability to track the location. That is the major problem facing with this convection treatment, okay, because the scientists, they are facing how to track the location. That is the major part, because in grade 4 kind of tumors, it's due to highly aggressive nature, okay, it penetrates the nearby cells. So always, I mean, within few days, okay, their locations, their spread is always changing. So that is the major part, okay, regarding the challenges regarding use of this convection-based approach. Now the next is the nucleic acid-based therapy. It's a very promising result because it direct benefit and it's a long-lasting therapeutic effect. But till date, despite their tremendous progress, only few nucleic acid-based drugs are being used, or that are part of this clinical studies, okay, or being tried on this different kind of cancer patients because of their some limitations. So what are those limitations, or what are those challenges that causes that limits the use of this nucleic acid-based treatment? That is instability problem. That is the major problem with instability can cause difficulty in traversing the biological barriers. That is the second problem. Now off-target effect and poor cellular uptake. So these are the problem with this nucleic acid-based treatment, okay, that must be resolved, okay, by any means, okay, so to enhance to use treatment of better outcomes. Because this is a good option, but because of these kind of problems, okay, still their use is very limited. Now, oncolytic viruses, it's a studies have reported the use of oncolytic viruses, okay, to target the brain tumors as a very promising treatment option, which is already tested. A few of the studies already has published. So many oncolytic viruses, after modifications, they are being used, okay, for the treatment of these kind of cancers, and they are the part of biotherapeutics, because after modifications, they are engineered to infect and selectively kill especially the cancer cells. So like paromavirus, okay, this herpes virus, adenovirus, okay, they are modified, and a lot of studies are published, okay, related to this, and they are giving the promising results. But the problem is that, again, how to change their pathogenicity and against which cancers, okay, it may be effective, that is still not clear. So studies are still going on, and they are using or working on this oncolytic viruses after modifications of these, okay, for as a treatment option for brain tumors. Now, in the current era of theranostic nanoparticles, so because this theranostic nanoparticles, okay, it utilizes the imaging techniques as well as the treatment, okay, at the same time, okay. So this is the giving very promising results. So can we modify, can we use the already discussed approaches, okay, within this theranostic nanoparticles? So it gives the better results, okay, rather than simply using theranostic nanoparticles, okay. So I think we can utilize the already discussed approaches, okay, with this theranostic nanoparticles. So why this is nowadays it's very popular, because the imaging options is available, as the option is also available by using this theranostic nanoparticles. So it's a great options, okay. Nowadays, okay, the people's researchers, they're working on this area, and it's could be nanoparticles used in personalized medicines or optimized treatment to each patient, taking into account the individual variability, because of variations in the patients as well grade, their age, their sexual types, okay, and their different physiological situations. So we can modify this theranostic nanoparticles, okay, for this particular set of patients, okay, and I think that is the part of this personalized medicines. Now coming to this conclusion part. So the poor prognosis and rapid recurrence of brain cancer cells are associated to its fast-growing process and invasive nature, which makes difficult the complete removal of the cancer-infected tissues. That is the bigger challenge, not only with the brain cancer, okay, it happens with the most of the solid tumors, which is very aggressive in nature, because till the time of surgery, or when, even if we give the chemotherapy before this surgery, okay, so it's very difficult to identify how much extent the tumor has spread it earlier, okay. So the organs after operations, okay, after surgery, there may be a residuals of those malignant cells, okay, they're still in that part. So that is very common, especially in the brain tumors, because the area is delicate, okay, each and every part is important for human being, okay. So how to identify which area has only which area becomes malignant, okay, that is the remains challenge, and could be resolved only with the imaging techniques by newer approaches. And theranostic imaging techniques, okay, could be option for exactly allocate the areas, okay, how much area has affected, especially for high grade tumors. Now the second conclusion is the heterogeneity of brain cancer within and between patient demands a patient focused method of treatment. That is the personalized medicine that we already discussed over here. So in patient focused medicine or personalized medicines, okay, the heterogeneity, different kind of expressions of this brain tumors in different populations, okay, within the tumor as well as within between the patients, okay, it's still a challenge, because each and every patients, their physiological conditions are different, okay. The same tumors may behave differently, okay, in different set of patients. Now, third conclusion is the implementation of nanotechnology is an effective approach considering all anatomic issues of the brain, since it will potentially improve the brain drug distribution due to interaction between the barriers and nanoparticles. Now, the next is in the current era of theranostic techniques, the nanoparticles are giving very promising results, okay, about the application due to their versatility to integrate multiple functional components in one system. Now, to improve theranostic approach, different active strategies can be used to modulate the surface of the theranostic particle, and also can take advantage of the characteristics of the microenvironment. Okay, that part is very important, because when the cancer grows or around this atmosphere that is known as microenvironment. Okay, so that means approach what is the pH of that area. Okay, what changes has happened in that area. Okay, that is very important to identify. Okay, for better treatment options. So that is all about this different approaches. Okay, being used for this brain cancer patient. Okay, again the problem, the issue with this brain cancer is the survival of those patients. How we can prolong the survival? Okay, so without a focused target, without harming the nearby cells, because it's related to brain and each and every inch or means centimeter important. Okay, because each and every part is resembles some functions. Okay, so we have to protect those parts. Okay, by using targeted approach. So thank you all. Okay, and patient to listening. Thank you.
Sir,
Now
thank you
you can
so much.
ask the questions, please.
Sir, thank you so much. Now this is a time for asking question. If anyone have any question, please put it to the chat box, or you can directly ask. Sir, related to my topic, when I was working in the project, I have one question. When I was working in the project, so as you already discussed in the presentation, whenever we are having a nucleic acid kind of receptor targeting strategy, so what exactly happening? The macrophage engulfing the propeptides. So in that
Yes.
case, what we can do, how to target brain cell effectively?
Especially with nucleic acid.
Yes, yes.
No, why we are not combining the other treatment option, okay, along with this approach, okay, because the nucleic acid is a very targeted approach. Okay,
Yes, yes.
so it's a very targeted approach. Okay, so we can't modify it, means this. We have to identify this, means to which receptor we are going to bind with. Okay, that means that nanomedicines. Okay, based on this nucleic acid approach. So I think means I think imaging part, okay, that is very important, okay, especially means you can means first identify, okay, the area which is involved, okay.
Yes, sir. Hello, sir. Am I audible? Hello. Yes, sir. Yes.
Hello,
Hello.
Dr. Rahul, I'm audible now.
Hello, sir. Can you hear me, sir?
Hello.
Sir, can you hear me?
Yes, yes. Ma'am. Okay.
Yes, yes, yes.
Yes, sir.
So, sir, thank you so much. I think you made it very clear. So, during the presentation, I think sir has discussed so many challenges related to brain cancer, emphasize on the triple B and blood-brain tumor barrier, and the opportunity to treat brain cancer also. Sir has given has completed all the aspects are related to biological or the brain cancer treatment. Sir, thank you so much for sparing your time.
Thank you.
So we are sure we will learn so many things and we will implement in our research work. So now, as we approach the conclusion of today's one-day workshop, I, Dr. Rahul Pratap Singh, Associate Professor and Principal Investigator at the Department of Pharmacy, School of Medical Life Sciences, I express our collective gratitude to everyone present here. Hello, am I audible? Hello, am I audible, sir?
Yes, you're audible.
Yeah,
You're
sir.
audible.
Thank you so much, sir. Now, in the conclusion part, on the behalf of just give me 1 minute, sir. There is internet glitch. On the behalf of School of Medical Life Sciences at GT Goenka University, I extend my heartfelt gratitude to our esteemed guests, distinguished speakers, and all participants. I would like to express my sincere thanks to Science and Engineering Research Board for their generous funding support through the project under the Empowerment and Equity Opportunity for Excellence in Science scheme. I am also grateful to GT Goenka University for providing the opportunity for the successful implementation of our research activities aimed at benefiting brain cancer patients. A special thanks to Vice Chancellor Professor B.S. Satyanarayan sir and Registrar Professor Dhirendra Singh Parihar sir, for their unwavering support during the project implementation. I appreciate the support of Dean Professor Sahil Arora for allowing me to explore my skill in the field of cancer nanotechnology and provided the necessary support to complete our project work. Now, this is the time to discuss about the workshop, the concept of dual receptor targeted nanomedicine, which can target two different receptors in brain cancer cells simultaneously, which hold immense promising in enhancing drug delivery specificity and efficacy. This workshop aimed to disseminate knowledge about advanced drug delivery system addressing challenges such as control release, targeting, biocompatibility, scalability, regulatory approval, and many more. Our goal is to ensure the effectiveness and safety of these systems in cancer therapy, aligning with sustainable development goals. The workshop is the workshop has covered critical topic including development and characterization of nanotheranostics system, application of QBD principle in pharmaceutical formulations, regulatory aspects of nanomedicine, predictors of outcome in brain cancer treatment. I am proud to report the progress of our project, including the guidance of 4 PhD research scholars and the support provided to Ms. Mehma, Ms. Bhavana, and Mr. Saurav Sekhar, who are near completion of their PhD research work. Additionally, I have guided more than 8 MPharm research scholars facilitating their research activities in my lab. Noteworthy achievement for our funded research project include guided PhD scholars, 2 scholars are nearly to completion by December 2023, and guided 5 research scholars, MPharm research scholars who got their degree, and recently I am guided 3 MPharm research scholars. And total publication from this project is 12, 8 research article and 4 editorial and review article. Total patent has been filed at number 5, and out of 5, 3 are under examination. Our distinguished speakers, Dr. MS Muthu, Dr. Datta Paude, Dr. Vikas Zawat, and Professor Ashok Kumar Gupta, sir, have provided invaluable insight during this workshop. Their expertise on nanotheranostics, QVD principle, regulatory aspect, and brain cancer management have enriched our knowledge. Once again, I sincerely thank all the speakers for sparing their wisdom and time with us. Our research inputs will undoubtedly contribute to our ongoing work. I also want to acknowledge the active participation and engagement of our audience, which have made this event dynamic and successful. We look forward to future collaboration and assignment with you all. Thank you once more for your support and dedication. Thank you, sir.